Quest Vision. To find ideas that become solutions to the world's most pressing concerns. Innovation and creativity are the sparks that ignite to transform lives and society. And these need space to grow.
We are home to these creative and innovative sparks. Our work goes beyond the disciplines, across our 9 faculties, 7 campuses, and being one of Africa's top business schools. As an African global university, we stand proudly among the world's top universities. We remain at the forefront of universal discoveries and provide authentically African solutions to the world's greatest challenges and opportunities. We are pioneers in our fields, from the humanities to cutting-edge technologies, continually seeking ways to improve the world around us. Our hybrid learning systems provide quality education that enables graduates to become adaptable game changers. We have molded nearly 500,000 alumni who have made their mark locally and internationally, and we continue to inspire and equip our students to uphold our firmly rooted culture of excellence. At the heart of what we do is our people. They are creative, critical thinkers who can shape society for the better, because that is how we teach, learn, innovate, impact, and live. We are the University of Pretoria.
Good morning, everyone. I'm very honored to open this conference, and I just wanted to thank everybody who's here, especially our international collaborators and partners who came from all over the world. I also would like to thank everyone who comes within the country and the African region who's here. I'm very happy that we'll be sharing the stage with the international players to learn from them, and so that we can tackle cancer together. Actually, in Africa and South Africa, we're very genetically diverse, so it's very important for us to be involved in cancer research and be able to come up with African solutions, but with the help of the experts, of course. And we're very happy that we have a lot of international speakers, world-renowned scientists, which I'm very honored as me standing here that they have descended from all over the world. I just wanted to say a little bit about the Pan-African Cancer Research Institute, which is a small history. Actually, it started in 2019 when the University of Pretoria actually approached me to come and start something in cancer research in the university. For me, it was exciting because University of Pretoria is one of the top 5 universities. So in other languages, is an Ivy League. So while it wasn't the first that I was working for an Ivy League university here, but just that I haven't been approached. I always applied and went for an interview, but this time I didn't. So in 2019, I started. There was nothing when I came, it was just me. So I started to go around and present my vision for cancer research. And then I came up with a strategy. Actually, I wrote the whole document that you see, but with the help of the partners and collaborators, they then looked at it, gave me input, then it was really improved. And then after that, I had to write all the documents that for approval through council, and then there was COVID after that. So everything slowed down, except that the university did approve funding for the state-of-the-art laboratories, but they weren't going to build them. They only started last year in 2022 to build, and they finished in December. So we are slowly trying to move in there. So a very big thank you also to the University of Pretoria for having the vision of driving cancer research, and also for going out there to look for me, which they got. And I think I've done a great job input, because the institute as such, we have a whole lot of scientists, basic scientists, we have clinicians, and which we are working together with. We've also now having satellites, University of Witwatersrand, University of KwaZulu-Natal, and now recently University of Cape Town. We already also have University of Limpopo, Sefako Mahato Health Science University. We have colleagues from other universities such as UNISA, University of South Africa, University of Johannesburg, and Walter Sisulu University. So it's growing, and I must say next year we are planning to move, because I think this venue is going to get smaller, to Cape Town. So I'm really thankful that you all descended here, and I'm make this an opportunity for you to interact with the best and collaborate more, so that we take this forward. We really strengthen cancer research. This is our first Pan-African Cancer Research Institute International Cancer Meeting, and we're gonna have every year one of those. Actually, we must be thankful that we're getting top scientists coming here to listen to them and interacting with them. When I wanted to listen to top scientists, I had to fly like more than 18 or 17 hours. I must go to the US, I must go to the UK, I must go to everywhere. We must make it in such a way that here it's a stage where we like, they descend to us and they come, they are happy to come. It's just that they needed a stage where they can be called. So when you call them, they come. And I think I'm very much in love with cancer researchers, because they're all over the world. They want to help. They defeating cancer together is a slogan of everybody. So with all those words, I would like to declare this conference open.
Thank you very much, Prof. Dlamini. It's a pleasure to be here. My name's David Bates from the University of Nottingham. This is Dr. Tula Molefe from...
I'm Dr. Tula Molefe from the Department of Medical Oncology at Steve Biko Academic and University of Pretoria, and a member of the Pan-African Cancer Research Institute.
Thank you. It's been an amazing journey watching the development of PACRI over the last 3-4 years, and hopefully helping to contribute towards it in some way. It's great that this year we've got so many people coming to visit. This first session is titled Diagnostics, and in particular what we're going to be doing is looking at the uniqueness of cancers in different parts of the world, and in particular in different parts of Africa, but also drawing analogies with other parts of the world where specific cancers have different characteristics from where most of the research has been done in the European Union and in the US, and why it's important to be able to do the research in the place where those cancers are actually the most impactful. So our first, so we have two speakers from the University of Nottingham. So obviously I should not be introducing them or asking questions about them. So I'm going to hand over to Dr. Molefe to introduce the first two speakers.
So our first speaker is Professor Kevin Sciences and Director of Undergraduate Courses in Cancer Sciences at the University of Nottingham. He received his BSc in Biological Sciences in 1986, followed by a PhD at the University of Birmingham in 1989. He did a postdoctoral work in France, followed by cancer research work in UK in London. He was appointed a lecturer before joining the University of Nottingham. And today his talk will be going through protein kinase inhibition in prostate cancer. Professor Gaston.
Good morning, everybody, and thank you for the invitation to the organizers. Thank you very much for the invitation to come and speak here. I promised, I know this is a session on diagnostics. I will mention diagnostics at least once. I'll try to mention it once. And it's also, obviously, I want to talk a little bit about the title of my talk. But what I should have really titled this talk was The Road to Exploring Kinase Inhibition, because I want to talk about how we got to this point, the point of what we're doing now. What we're doing in my lab now is that we're looking at kinase inhibition in prostate cancer, and we're also helping to establish a Kenyan biobank. But that's really what we're doing precisely now. What I'm going to talk about in this talk is the road to getting there. I'm very happy to come to Pretoria because, guess what? I was born in Pretoria, but unfortunately not this Pretoria. I was born in Pretoria in Newcastle upon Tyne, which is not quite the same as this Pretoria. And that's the street where I was born. And it's in black and white because it was a long time ago. There's no color photographs of that. So this is an overview of what I'm going to talk about today. I'm going to talk mostly about a transcription factor called PRH-HEX, which I was introduced to this transcription factor by Professor Jayaraman, and she's going to tell you more about it in her talk. But I've studied how it works in the context of different types of cancer, and we've looked at how this transcription factor controls cancer cell biology. So it controls cancer cell proliferation, cancer cell migration, and also cancer cell invasion. And it itself is regulated by a kinase called protein kinase CK2, and I'll talk about how CK2 controls PRH in this talk. And we've looked at that and studied this transcription factor in two different contexts. The first is cancer, and the second is intimal thickening. I'm not going to talk about intimal thickening today, but if anybody's got wants to talk about that, please talk to me afterwards. So just a very brief introduction. So the PRH stands for protein-rich homodomain protein, and the protein is also called HEX, or hematopoietically expressed homeobox. We don't like to use that title of HEX because this protein is not just expressed in the hematopoietic system, it's expressed in just about every cell that we've ever looked at. And it controls cell migration and cell invasion. Protein kinase CK2 is you might have heard of before. It used to be called casein kinase, but it's a ubiquitously expressed protein kinase that responds to cell stress and other growth factor signaling pathways, and then changes the activity of lots of things in the cell. And CK2 activity, generally speaking, goes up in cancer cells, and that's why we got interested in it, and in some other disease states as well, such as intimal thickening. And I'm going to tell what the take-home message from my talk is that phosphorylation of PRH by CK2 is important in tumorigenesis and in intimal thickening. So the reason that I got interested in prostate cancer was because we were looking at what happens to the PRH gene in prostate cancer cells. So in the top of this diagram here, it shows you each bar represents a different patient, and the genes here, it represents whether the genes are deleted or not. So for example, you can see that a gene called P10, and by the blue bars indicates a deletion, you can see that P10 is very frequently deleted in prostate cancer cells. And that's been very well established. And also the c-MYC protein, which stimulates cell proliferation, is often amplified in prostate cancer cells. But just at the very top there, we have PRH-HEX. And you can see that the PRH-HEX gene is frequently deleted in prostate cancer cells. And that's one of the reasons that I got interested in looking at prostate cancer and seeing what PRH was doing in these cells. Also, it turns out, if you can see in the bottom graph there, that the gene that encodes PRH is often highly methylated in prostate cancer cells, and that increased methylation turns the gene off, and so it reduces PRH expression. So we started off by looking and seeing what was happening to PRH. So we took some antibodies that recognize the PRH protein, and we stained prostate cancer tissues. So I'm not going to go through all the details of this, but suffice to say that in normal cells, we could see PRH staining for the PRH protein, and also in I've got a laser beam. Also in benign hyperplasias, we could see PRH staining, and also in prostate adenocarcinomas, we could see PRH staining. So the protein is expressed in prostate cancer, both in the pre-cancer state, the normal state, and in cancer cells. But interestingly, the amount of protein staining in those cells goes down as we head towards cancer cells. We could see that more clearly if we stained a TMA with lots and lots of samples. And what we could see there was that as the Gleason score increases, then the amount of PRH staining decreases. So this thing is going away in prostate cancer cells. So that led us to investigate, well, what is it doing in these prostate cancer cells? To do that, we have two different model systems. We have these PNT2C2 cells, which are a normal immortalized prostate epithelial cells. And we also have prostate cancer cells, such as PC3 and DE145 and other prostate cancer cell lines. So the first thing we did was to knock down the PRH protein in these normal prostate epithelial cells. And what happens there, if we're measuring, for example, chemotaxis or cell migration in response to a chemical stimulus, you can see that in the knockdown cells, which are in gray bars here, we see increased cell migration in response to this signal. Conversely, if we overexpress the PRH protein in prostate cancer cells, and remember in prostate cancer cells, they generally speaking have lower PRH activity or lower PRH staining. If we overexpress PRH in these cells, then we see a decrease in cell invasion. But it also controls cell migration. As well as controlling cell migration, it also controls cell proliferation. So to look at that, we take our knockdown cells again, and then look for their proliferation using, in this case, BRDU incorporation, which is an assay of cell proliferation. And you can see that the knockdown cells are much more highly proliferative. And if we do the converse experiment and overexpress PRH in these prostate cancer cell lines, like D145s, then you see that there's in the overexpressing cells here, which are with an adenovirus that makes PRH, then when we overexpress PRH, then their cell number is reduced. So PRH is controlling cell proliferation and cell migration. More recently, we've started to use an inducible system to overexpress PRH in different cells, so we can just turn on its expression using doxycycline. So we have a doxycycline inducible promoter that turns on PRH expression. So you can see here, here's the MIC-tagged PRH protein. In the absence of doxycycline, we don't see it. We add some doxycycline, and then we induce the protein expression. And you can see here that when we induce with doxycycline, if you look at cumulative cell number, it's strongly reduced by PRH expression. And again, the proliferation is decreased. So of course, we wanted to know what genes PRH is regulating in these cells. And I'm not going to go into details because it's not that surprising. It turns out that when we do RNA-seq to look at the genes that are being regulated by PRH, it turns out that it's virtually everything to do with cell proliferation and cell migration. So the top group down at the bottom here is E2F targets. E2F is a protein that stimulates the cell cycle. And you can see that all these E2F genes are being downregulated by PRH expression. So switching now to protein kinase CK2, how do we get interested in that? So I hope I've convinced you that PRH is controlling the proliferation and migration of prostate cancer cells. So what we wanted to do was then say, well, okay, well, how is it doing that? How is it controlling these cells? Let's see if we can find proteins that interact with PRH, and maybe that will give us a clue as to how it functions. So we did a very old technique, which I love still, which is that we screened a yeast-2 hybrid library. And what happened is that we found some PRH interacting proteins. So in this experiment, we have the GAL4 DNA binding domain fused to PRH, and then we screen a library that has lots of different cDNAs connected up to the GAL4 activation domain. And we isolated cDNA that encoded a protein that enabled this complex to activate transcription. And it turned out that that was CK2 beta, the beta subunit of the CK2 protein. So CK2 beta binds to PRH. So this is not the same as phosphorylating. I'll talk about that later. This is just a binding event. So this kinase regulatory subunit, so it's not the part that does the phosphorylating, it's the part that regulates the kinase, and that binds to PRH. I won't go through this in detail, but just to show basically that if we do pull-down assays, for example, we can pull down CK2 from cells with PRH, and we can do the reverse, we can do co-IPs, but I won't go into details. So what does CK2 do to PRH? Well, to look at that, what we did was we took the purified PRH protein, and this is a DNA binding transcription factor, so we can measure its binding to DNA. This assay is called an EMSA, where we look at the binding to DNA down here. We separate the bound DNA and the free DNA using a polyacrylamide gel. Here's the free DNA, and you can see that when we add PRH protein, we get a retarded complex. Then we can add CK2. If we add CK2 alone, which is in this one, if we add CK2 alone, nothing really happens. The PRH protein is still binding to DNA. But if we add CK2 and ATP, which is the substrate for the kinase, then we see that there's a complete block of PRH binding to DNA. So we inhibit the binding of this protein to DNA using by CK2, presumably by phosphorylation. We can show it is that because if we then add an enzyme called CAP, or calf alkaline phosphatase, that will then remove those phosphate groups. If we do that, then the DNA binding activity comes back. So this is a reversible switch for DNA binding. If it gets phosphorylated, it can't bind to DNA. If it gets dephosphorylated, then it can bind to DNA again. So our next step was then to map these phosphorylation events and try and make sense of what was happening. So we isolated the phosphorylated protein. I won't go into details about how we did that. And we mapped where these phosphorylation events were occurring by mass spectrometry. And it turned out that it was occurring on serine 163 and serine 177, two serines within the PRH DNA binding domain. Both of these serines come very close to the DNA backbone. So in these models here, the protein is in blue and the DNA is in red. And you can see that serine 163 sits very close to the phosphodiester backbone of the DNA. So you can imagine that if you put a phosphate group here, it's going to be in direct opposition to the DNA backbone, and that will stop this protein from binding to DNA. Similarly, 177 is also close to the DNA, and if you phosphorylate there, the protein can't really bind to DNA at all. To prove that, or to try and investigate that, we made these mutants that I'll just digress for a minute to tell you about. So we have two different mutants. We have glutamic acid mutants. So we call that PRH-EE because it has glutamic acid at those two positions, serine 163, serine 177. And we also made this mutant called CC, which is a cysteine substitution, and that means it can't be phosphorylated by CK2 because you can't add a phosphate to a cysteine amino acid. And you can see here that the wild type will bind to DNA, and then if we mutate in these glutamic acids, now the protein can't bind to DNA very well, and if we mutate both to glutamic acid, it can't bind DNA at all. That allowed us to go and see, well, what happens in cells when will CK2 stop PRH from binding to DNA in cells? So what we did for this experiment is that we overexpressed either the full-length PRH protein or the full-length PRH protein with these different mutations, either the CC mutation or the EE mutation. And we measured its binding to a PRH target gene using quantitative CHIP, or chromatin immunoprecipitation. What you can see here is that if we look at the wild-type PRH protein, it binds to this target. This is actually the VEGF-R1 gene, which is a gene that Dave Bates turned us on to because he said, why don't you look at that and see if PRH is regulating it? That was a long time ago. But anyway, PRH binds to the VEGF-R1 gene, but and the control doesn't, which is this in the white bars. If we overexpress PRH and CK2, then we no longer see any binding to the VEGF-R1 gene. If we overexpress our CC mutant, remember this is the one that can't be phosphorylated, it binds to the VEGF-R1 promoter and regulates it. But now if we overexpress CK2, nothing happens. It can still bind to the VEGF-R1 promoter. And of course the EE mutant, which doesn't bind DNA, that doesn't regulate this promoter. And the phosphorylation, as you'd expect, if we then block the phosphorylation, that stops it from regulating cell migration as well. So in this experiment, we overexpress PRH. If we overexpress PRH, we get an inhibition of cell migration. If we overexpress PRH-CC, we inhibit the proliferation. But now, when we overexpress CK2, it can't block that inhibition of cell migration. And the same is true for cell invasion as well. So PRH will repress cell invasion, but if we overexpress CK2, it can no longer block that invasion. So obviously, we then wanted to go back and see, well, if we look in prostate cancer, does this have any relevance to what's happening in the cancer cells? Is PRH getting phosphorylated in the cancer cells? And of course, I wouldn't be telling you about this if it wasn't. So to do that, we had to make antibodies that recognize either the phospho-PRH, so these antibodies here used in this Western recognize phosphorylated PRH, whereas these antibodies up here are recognizing the unphosphorylated PRH protein. And we looked in, to begin with, in our immortalized prostate cancer cells and prostate cancer cells. You can see here that PRH is present in all three, but phosphorylated PRH appears to be much higher in the cancer cells. And we know this is CK2 dependent because if we add inhibitors like DMAT and TBB, which both inhibit CK2 activity, then they both prevent phosphorylation of the PRH protein. And when we stained our clinical samples, what we found was that the phosphoprotein is very highly enriched in benign prostatic hyperplasias. So as these cells become hyperproliferative, they express high levels of the phosphorylated PRH. So these first two columns here are the phospho antibodies, and red means high expression. So we're getting very high expression in these benign prostate samples. But in cancer cells, phosphorylated PRH staining seems to go down. And that seems to agree with our overall assessment that the unphosphorylated protein was going down in those cancer cells too. And it's not just in prostate cells. It's more widespread than that as well, because if we look, for example, in breast cancer cells, in ductal carcinoma in situ samples and normal breast samples as well, this is some work that was done by Professor Jayaraman and her group in Birmingham University when she was working there. And she took these antibodies that recognized the phosphorylated PRH and stained this collection of samples. And you can see that we get very high staining in low-grade DCIS, which is similar in many ways to benign prostatic hyperplasia. So in several different types of tumor cell, we're getting phosphorylation of the PRH protein, and then that would obviously be expected to inactivate the PRH protein and then allow those cells to proliferate more and also to migrate and invade. I said I would mention diagnostics. One thing we're trying to do is to try and use these antibodies as a way of trying to diagnose the very early stages of cancer transition. So they're not very useful for the late stage, because at that point, PRH staining is going right down. But they can really tell what is a benign lesion and what is where maybe they're the very early stages of transition. So I'll just digress for 1 second to tell you a little bit more about phosphorylation, mostly because I love this story. So it turns out that phosphorylation doesn't just stop the protein from working. It also induces protein processing by the proteasome. So in this experiment, what we've done is we've added an isomycin, which blocks translation. And then we've looked at PRH levels with time. So if we look at the hypophosphorylated PRH, and we add this anisomycin, and we separate the cells into either a post-nuclear fraction or a nuclear fraction, you can see that there's very little change in PRH levels. So this protein is very stable. We block translation, the protein stays there. But the phospho-PRH protein, which is shown by this antibody, here's the phosphorylated protein, you can see that it disappears with time. So it's a very unstable protein. But the interesting thing that happens is not that this phosphoprotein goes away. The interesting thing that happens is that down here we have this other protein, which is smaller than the full-length PRH by about 10 kilodaltons, and it increases when we add these inhibitors of translation. And so what we think happens is that this protein is getting processed by the proteasome and turning into a smaller version. We know it's the proteasome that's doing this cleavage because if we add MG132, which is an inhibitor of the proteasome, you can see now, in the presence of this proteasome inhibitor, the phosphoprotein here is now stable because there's a proteasome inhibitor present. So what happens is that the full-length protein is being cleaved by the proteasome to make a smaller phosphorylated, non-DNA binding version of the PRH protein. So why is that? Why am I telling you this? I'll get there. I'm telling you that because it speaks to how PRH regulates transcription. I haven't got time to go into the details because we published this a long time ago, that the PRH protein, one of the ways it can regulate transcription is that it binds to a co-repressor called TLE. This was discovered by Professor Jayaraman several years ago, and it regulates genes such as VEGFR1. But if we co-IP things that bind to TLE, the co-repressor, we can see that this endogenous PRH delta C, the phosphorylated protein which has lost its C terminus, it still binds to the co-regulator protein. So it binds and it sequesters this co-regulator. So the way it works is just summarized in this cartoon, which, as I said, I haven't shown you all the evidence because we published this quite a couple of years ago. But the PRH normally binds to DNA, recruits TLE, and then represses transcription. But if CK2 phosphorylates the protein, it then produces this phosphoprotein, which gets processed to make PRH delta C. And the PRH delta C can sequester TLE and stop PRH from repressing transcription, which I think is a fantastic mechanism for turning off genes. The reason that it's important is because, of course, when you phosphorylate proteins, we don't often think about this, but when you phosphorylate proteins, you might only phosphorylate 1 or 2% of the protein that's in a cell. So how do you turn a gene off? By phosphorylating 1 or 2% of the protein that's there. One way that you can do that is you can phosphorylate a tiny amount of the PRH that's in the cell. And what will happen is it will get processed and then it will start to compete with itself. So this is a way of saying you get a small amount of phosphorylation of PRH, you get a massive effect on transcription, which I think is a fantastic mechanism. And that's what got us into studying CK2. So I told you I'd get to CK2 inhibition eventually. So then, having worked out what CK2 is doing to PRH and what PRH is doing in prostate cancer, that's what led us to start studying these inhibitors of protein kinase CK2 in prostate cells. And what you can see is that inhibitors, this is one example, this is an inhibitor called CX4945. And as you add the inhibitor, then you strongly inhibit prostate cancer cell. This is viability. So prostate cell viability strongly inhibited by these inhibitors, as you would expect. This is a mouse prostate cancer model. DVL3 is a mouse prostate cancer cell. And you can see you get the same thing. You get strong inhibition. As you'd expect, the reason for this strong inhibition of cell viability, we think, is because we get a massive increase in apoptosis. So, for example, if we look at cleave caspase 3, you can see when we add the inhibitor, it's strongly induced. As you'd expect, when we add the inhibitor, phospho-PRH goes down, and the unphosphorylated PRH goes up, as you'd expect. So, but how much of this effect is due to PRH? Because it could be that the inhibitor is doing this by inhibiting lots of other things. We think that much of the effect of CK2 inhibition is going through PRH in these cells, at least. So that's shown in this experiment where we have our normal immortalized prostate cells. And if we add TBB, which is an inhibitor of CK2, you can see you get a strong reduction in cell number. But if we take cells where we've knocked down PRH, so PRH knockdown cells, so now these cells, they proliferate more rapidly than the control cells. So there's more cells. But now the inhibitor has no effect. So this inhibitor has very little effect on these normal immortalized prostate cancer cells, showing that much of the effect of PRH, of CK2 inhibition, is going via PRH. So you can see, as I summarize here, you've got PRH, it's repressing cell migration and proliferation. CK2 is controlling PRH, and we can add CK2 inhibitors, and they will block this step. So in other words, CK2 inhibitors increase the amount of PRH which is present within the cell, and then can control these events. So this is work. This is from not from my group. This is from a different group who have taken our PRH mutants and then looked to see what happens in a different kind of cancer. And the interesting thing here that they've shown is that the phosphorylated PRH is not just inactivating PRH, which is a repressor of proliferation. Phosphorylated PRH expression by itself can be oncogenic, at least in some cells. So they were looking in colorectal cells. And I won't go into great detail, but just to cut a long story short, when they overexpress the PRH protein, you get a small increase in tumor weight. But PRH-CC, which is our non-phosphorylatable, that doesn't increase tumor size at all. And PRH-EE, which is our phosphomutant of PRH, you can see that you get even larger tumor size. So in this case, in these colorectal cells, the phosphorylated protein is not just inhibiting the negative effects of PRH. In fact, in these cells, PRH seems to have a positive effect on tumor growth and tumor progression. And they showed very elegantly in this recent paper that the phosphorylated protein can't bind to DNA, but what it can do is that it can bind to these co-activators on the DNA and regulate gene expression. So just to summarize what I've told you, we seem to have a repressive complex, which is PRH bound to DNA with its co-repressor proteins, not just TLE and other things, and it's repressing cell migration and cell proliferation. We then phosphorylate PRH by CK2, and that stops this leg, stops the repression of proliferation, but also it allows PRH to bind to TEAD4 and other proteins, and then to act in a completely different way to activate cell proliferation and migration. So now we get to the new part. What we wanted to do was to study, well, okay, I hope I've convinced you that PRH controls cell proliferation and that CK2 inhibition can also prevent the proliferation of these cells. We wanted to know what signals were in tumors, what signals are causing this PRH phosphorylation. And of course, where the signals are coming from is the tumor microenvironment. One signal that we've looked at in detail is TGF-beta. TGF-beta produced by immune cells and other cells in the tumor microenvironment, when it's added to the cells, will reduce PRH, that will not affect PRH levels, will reduce PRH levels a bit, but it will, I can't really see now, will reduce, I don't think you can see it very well, but it will increase PRH phosphorylation levels. I don't know if you can see that very well, but it increases PRH phosphor levels and decreases PRH levels. So signals from the tumor microenvironment are controlling PRH. And so we decided to team up with a great group in Kenya to look at and to enable us to compare the tumor microenvironment in UK samples and in Kenyan prostate cancer samples. So we teamed up with Dr. Veronica Manduku at KEMRI in Nairobi, and her team is investigating the PRH tumor microenvironment and staining for PRH and staining for markers of infection and immune cells. And our team is doing the same, but on UK samples. And it's a great collaboration because it allows us to compare the different immunogenic signals that are occurring in her samples in Kenya and our samples in the UK. We started this just before COVID, and then guess what happened? So COVID happened, and we haven't really progressed this very far. So hopefully, if I'm lucky enough to come back again next year or the year after, I'll be able to tell you the outcomes of this research. But at the moment, we've just got a study which is ongoing, looking at PRH, the signals that control PRH in prostate cancer cells. So just to conclude, I've told you that PRH inhibits prostate cancer cell proliferation, at least in the context of the prostate, but it can have different effects in different cell types. And phosphorylated or inactivated PRH is increased in some tissues, like, for example, benign hyperplasia in the prostate and also in DCIS. And the phosphorylation prevents it from repressing. But in other cell types, the phosphorylated PRH is probably oncogenic. It certainly appears to be in colon cancer, and we think it might be in other cancer cell types as well. And we think that the effects of PRH, of CK2 inhibitors are very, very strong, but we think that much of those effects are mediated by PRH levels, because if we don't have PRH, then the CK2 inhibitors don't have very much of an effect. So I'm finished there, and I just want to say thank you to the people who did the work. This is all a long-standing collaboration with Sheila Jayaraman, who's going to speak next. She was at the University of Birmingham when we started this collaboration. She's now at the University of Nottingham. Sorry, I should have changed that. And I'm also collaborating with Veronica Manduku and her team at KEMRI to look at PRH in African samples. And that work is, and much of this work that I've shown you is funded by the UK Medical Research Council. And the people in the lab who did the work are shown in bold here, mostly Yudma and Yusra, and also Eric more recently, and Anya are down at the bottom here as well. And thank you for your attention.
Thank you, Professor Gaston, for an informative talk. We'll be taking questions later once everyone has spoken, just for the sake of time. Our next speaker is Professor Padma Sheila Jayaraman. She's a professor of cancer biology and gene expression at the University of Nottingham, and also teaches epigenetic and cancer therapeutic modules in the cancer sciences. She's a research lead and co-director of the Center for Cancer Sciences and the theme lead for Defeating Cancer in Biodiscovery Institute She's also involved in research knowledge exchange locally and internationally. Her research is aimed at looking at PRH-Higgs, and she'll be going through us with talk on cholangiocarcinoma.
Hello, everybody. Can you hear me? Great. First, I'd like to start by thanking the organisers for inviting me to speak here. It's a real privilege to be able to come here and tell you about my research. So I'm going to talk to you about a cancer that's not very well known or understood very well, and that's cholangiocarcinoma. I'm going to divide my talk up into 3 sections. I'm going to tell you a bit about cholangiocarcinoma generally, and then I'm going to talk to you about PRH in cholangiocarcinoma, and then just very briefly at the end, I'll touch upon some current experiments that we are performing on the area of screening repurposed drugs. Okay, so what is cholangiocarcinoma? Well, it's a cancer of the biliary tree, and it's considered to be a fairly rare cancer, and I'll touch upon that again in a minute. But worldwide, it makes up approximately, it's reported to make up approximately 3% of all GI tract cancers and 10 to 20% of all liver cancers. But I'll come back to that point in a minute. So just to explain exactly what the biliary tree is, try and use the laser pointer. So the biliary tree extends throughout the liver, and it's made up of a network of ducts and ductules that permeate the entire liver, and then the ducts and ductules come together to form the common hepatic ducts, the larger ducts, and they extend just outside the liver, and then they extend to the gallbladder and down to a very large duct known as the common bile duct. And the purpose of the biliary tree is to transport bile made by the hepatocytes in the liver to the gallbladder for storage, and then release from the gallbladder when required to the duodenum when bile is required for digestion. So there are 3 forms of cholangiocarcinoma. There is the intrahepatic cholangiocarcinomas that occur obviously within the liver proper. There are the perihilar cholangiocarcinomas that occur just outside of the liver. And then there are the distal cholangiocarcinomas that occur in the distal region from the liver. And the important thing to note is that the 3 different subtypes are quite distinct. They've got different epidemiologies, pathobiologies, and management. Cholangiocarcinoma as a whole is the second most common primary liver tumour following hepatocellular carcinoma, and it is common in people in their 70s, so more elderly people, and more common in males than in females. And the intrahepatic form of cholangiocarcinoma actually makes up only a minority of all cholangiocarcinomas, but it is one of the most deadly and more aggressive forms of cholangiocarcinoma. And the 5-year survival rate for this form of cholangiocarcinoma is really very dismal and is less than 5%. And often people present very late, and actually the mortality is very high. People often only last a few months, in fact. So although I told you that the prevalence of cholangiocarcinoma, or the incidence, is about 10 to 20% of all liver cancer, a recent study carried out by Professor Zahid Khan at Imperial University found that, noted that liver cancer had increased massively in England alone, and that there had been 62,000 cases of primary liver cancer between 1997 and 2016. And interestingly, he reported that there are, in fact, 38% of which were intrahepatic cholangiocarcinoma. So it seems to be that there may be very region-specific differences in incidence of cholangiocarcinoma, and it might be much more underreported than we originally anticipated. When we look at the mortality rates, we can see that there has been a global trend across many countries for the mortality to be increasing for intrahepatic cholangiocarcinoma. And this is evidence over in the 90s, but this is still occurring even now. And a recent review pointed out that in the USA, there's been a notable increase in mortality, and perhaps of quite considerable interest is that the African American individuals have been, the incidence is higher in African American individuals than in other groups. So I said that cholangiocarcinoma is a relatively rare cancer globally, and at least in the global north, and that means that the incidence is about 6 cases per 100,000 of the population. But in some parts of the world, such as in Southeast Asia, shown here in red, the incidence is markedly increased, and it is increased right up to 85 per 100,000. And I'll explain why in a moment. But there is very little information for the incidence of cholangiocarcinoma in Africa as a whole. However, if we look for mortality rates for liver cancer in Africa or globally, but just focus on Africa for the time being, we can see that mortality for liver cancer is quite high in certain regions of Africa, and in particular in West Africa. So based on our understanding that globally what the percentage of cholangiocarcinoma is maybe higher than was originally reported, I think we can reasonably speculate that there might be quite considerable mortality arising from cholangiocarcinoma in Africa too, in certain regions of Africa, I should say, as well. So what are the risk factors for cholangiocarcinoma? Well, the major risk factor that is really well established is the presence of infection with liver flukes. So the flukes are shown here, and they are known to occur to infest the fish that are present in the rivers in certain regions of Southeast Asia, and in particular in northeastern Thailand. And the liver flukes live within the fish and then are consumed by people in these regions, and they have the practice of not cooking the fish, so they eat a lot of raw fish, and this allows the liver flukes to survive, and the liver flukes survive in the bile ducts of people for many, many years, and they essentially cause a massive chronic inflammatory reaction within the people of those individuals that then increases the incidence of cholangiocarcinoma to very high extents. So I think that speaks to telling us how important the environmental exposures are for the incidence of cholangiocarcinoma. The other things that are important risk factors are inflammatory conditions, and these are conditions that are commonly found in the West. Cholangitis is a major risk factor for cholangiocarcinoma. Cysts, toxins, bile duct stones, and hepatitis B and C virus infection, and lastly, alcohol consumption. One thing I thought I might just highlight is a recent study that was performed by a group from China, and they showed that in intrahepatic cholangiocarcinoma patients, that there is a genomic aflatoxin signature that is found, and aristocolic acid signature that is found only in the Chinese population, but not in Western populations, telling us that it's very important to look in different populations for different mutagens. So these aflatoxins are aristocolic acid mutational signatures presumably come from environmental exposure to those toxins in the diet. Okay, so there's a very urgent need for new therapies for cholangiocarcinoma, and this is because this particular disease is generally very asymptomatic. The is a huge lack of good biomarkers for cholangiocarcinoma. And because it's mostly asymptomatic, there's a very late diagnosis, and this occurs often after metastasis has occurred. And therefore, the majority of people that get cholangiocarcinoma do not get offered resection. They are not able to be resected. The majority of people have to be treated either palliatively or with chemotherapy. And although there are some now modest improvements in the treatments with chemotherapy, for example, there is the durvalumab, which is a PD-1 inhibitor in immunotherapy, which has been added to GemCis. This only makes a modest difference in treatment effect. So there's an urgent need for new treatments, and I'll talk about the second-line treatments in a minute. I just wanted to also tell you about the sequencing that has been carried out for cholangiocarcinoma. Whole exome sequencing has shown that there are a very large number of mutational events that are associated with cholangiocarcinoma. In fact, up to 40% of cholangiocarcinoma cases are associated with mutation, and many of those are potentially targetable. However, the problem is that those mutations don't occur in very many people. Although there are very many mutations that have been discovered, the prevalence is that they're only in very few individuals. So the best or the most common mutations are things like the FGFR2 rearrangements and the IDH1 mutation, which is a mutation of a metabolic enzyme that affects DNA methylation. These two events still only occur in 15 to 20% of intrahepatic cholangiocarcinoma patients. And although treatments have been developed for these rearrangements and mutations, still that means that 80% at any one time cannot receive treatment. And so it is still really very important to develop new treatments for this disease. So that brings me on to PRH in cholangiocarcinoma. So Kevin has kindly introduced the PRH protein. I come at the protein from many years ago when I was very interested in understanding how did it work as a transcription factor. And we know that it's part of the homeodomain class of transcription factors that is responsible for cell fate. And my group has been very interested in understanding how it binds to DNA and brings about gene regulation. And our lab has spent many years trying to understand how this protein, which actually forms homooligomers, that on DNA can form homooligomers, are able to integrate developmental cues, proliferative cues, growth regulatory cues, and bring about changes in gene regulation. And if anybody's interested in that, I can talk to them about that later on. But for the purposes of this talk, I just want to tell you that PRH is a really important regulator of liver development and bile duct development. And in fact, it's important for both the production of the hepatocyte lineage and the bile duct lineage, and is involved in regulating the expression of these lineage-specific transcription factors. Knockout experiments of PRH or HHEX cause loss of liver development completely or loss of bile duct development, depending on when the knockout was introduced. So it's an important regulator for liver and bile duct development. And contrary to what Kevin has shown you, that it's a tumor suppressor in prostate and breast cancer, we showed a few years ago that PRH is a really important oncoprotein in cholangiocarcinoma. So it's driving the growth of the cancer. We saw in RNA-seq, the TCGA cohort, the RNA-seq database, that PRH mRNA levels are very high in the intrahepatic cholangiocarcinoma patients. We look to see a protein level and see that it's high in protein in a tumor microarray compared to normal bile duct cells. And when we carried out xenograft experiments, we saw that when we knocked down the PRH protein, we couldn't form xenografts. They formed at a very low rate, and they didn't grow very well. So that gave us evidence that PRH is an important oncoprotein in this tumor type. And when we looked to see how is it regulating the growth of the tumor cells, we could see that if we overexpress the protein, the tumor cells are proliferating faster. If we knock down the protein, here's the knockdown, the tumor cells grow slower, but we're not having any effect on apoptosis. We did a lot of transcriptomics and did gene ontology following RNA sequencing. And we see that, as expected, we can see many genes that are involved in the regulation of cell proliferation have been changed, whether we carry out knockdown or we carry out overexpression. And indeed, quite interestingly, Wnt signaling pathway genes are strongly changed as well. So we just did a little bit of validation and showed that several cell cycle genes are altered in their expression, particularly cyclin D2, which is activated by PRH expression. Here's a knockdown experiment, and you can see cyclin D2 goes down. Interestingly, cell cycle inhibitor genes, so those are CDKN1B and 2B, were de-repressed when we knocked down PRH. And we also carried out chip sequencing experiments to show that the genes are bound by PRH when they're regulated. So the cell cycle inhibitors that are repressed by PRH are directly regulated, and it's bound near the gene. And we carried out similar experiments for many of the genes in the Wnt signaling pathways. Okay, so what we found by doing all of this work was PRH is altering the cell cycle, and it's doing this by a number of mechanisms. It's repressing cell cycle inhibitors, so obviously the cells can cycle faster. It's promoting the expression of cyclin D2, which is a ligand for CDK4/6, and it can also increase the activity of multiple Wnt pathway genes indirectly and directly. So altogether, it's going to affect the cell cycle very potently. So we speculated that, or hypothesized, that perhaps PRH high cells would have very fast cell cycle, and therefore PRH high cells might well be more sensitive to cell cycle inhibitors than other cells. To test that hypothesis, we tried to test what the effect of palbociclib would be on PRH high cells. And we chose palbociclib because palbociclib is a well-known CD4/6 kinase inhibitor, and it's a nice approved therapy. So it's an approved therapy for metastatic ER-positive breast cancer, and we know that it has quite tolerable side effects in people. So we thought this would be quite a useful therapy to be transferred to for the use of cholangiocarcinoma patients. So to cut a long story short, we tested lots of cholangiocarcinoma cell lines, and we overexpressed PRH in those cells, and we see that in each case, having more PRH in the cell seemed to increase the sensitivity to the drug. But obviously, we're not going to actually put PRH into people's cells. What we were thinking was that maybe we could use PRH as a marker for determining who would be useful to target with that particular therapy. We also needed to check what happened if we knock down PRH, and you can see here that the knockdown cells are less sensitive to this particular inhibitor. So it looks like we are getting effects through palbociclib through PRH. And you can see this again in this experiment. Here are CCLP cholangiocarcinoma cells treated with palbociclib. There is an effect of palbociclib, But if we put more PRH in the cell using an adenovirus and then treat with palbociclib, you can see that the effect is much, much greater, partly because we've stimulated the cell cycle. But okay, that's fine. Palbociclib seems to be quite useful. But we were interested to see another effect of palbociclib, which is that in these cells, it's not just stopping the cell cycle, it's also increasing caspase-3 activity, which is a marker for apoptosis. So the palbociclib is increasing apoptosis in this cell. And more interestingly, considering that PRH itself does not have an effect on apoptosis, we have seen that the presence of PRH in the cells increases the effects of palbociclib on cells, on the apoptotic nature of the cells. And we see this in a number of different cell lines, that in each case, having more PRH in the cells potentiates the activity of palbociclib. You can see the difference here. And just to make sure that we're actually seeing apoptosis and not just seeing an increase in caspase-3, we carried out the experiment using annexin staining, and we can see that yes, when we put palbociclib in the cells, that's in these two columns, we have more apoptosis, but when we put palbociclib in the presence of PRH, we have even greater levels of apoptosis. Okay, so that's great. Palbociclib does seem to be useful in PRH high cells, and many tumor cells do have high PRH, and PRH is pushing the tumor to grow. So it might be a useful treatment for some types of cholangiocarcinoma. But what we were interested to do next were to find out if there were other drugs that might work with palbociclib to treat cholangiocarcinoma. And the reason for this was there had been a basket trial carried out a few years earlier, where they showed that palbociclib monotherapy was not that effective in treating bile duct cells and pancreatic cancer cells. I should say we need to look at this trial with caution, because it's a very, very small trial of only 10 patients with biliary tract cancer. However, it made us think that maybe we need to think about other treatments as well, not just palbociclib. So we decided that a good thing to do would be to try screening for the effect of repurposed drugs on cholangiocarcinoma. So these are drugs that are commonly used in man to treat other conditions. So we already know that they're not highly toxic in man, and we would use concentrations that are less than what would be used to treat these other conditions, and see if any of those known inhibitors or known drugs could kill tumor cells. And we screened a large panel of drugs with the help of Dr. Farhat Kanem, who at the University of Birmingham, and we identified a number of drugs that can kill cholangiocarcinoma cells on their own. So that was great. And we identified one particular drug, which had good effects on cell viability. So it reduced cell viability, it decreased proliferation, and also itself increased caspase-3 activity, suggesting that it could also increase apoptosis. So the final question that we wanted to ask was, would they work together and in combination well? And we carried out Chu-Talay combination index experiments. And what you see is that palbociclib has a reasonable effect on reducing the proliferation of these cells, or the viability of these cells, I should say, not just the proliferation. Drug 56 has a reasonable effect, but putting these two drugs in combination at a variety of concentrations always has a better effect. And the combination index was less than 1, which suggests that these two drugs in combination might well be synergistic. So in summary, what I've told you about is that the prevalence of cholangiocarcinoma might be higher than we expect, that it's an PRH is a transcription factor and an oncoprotein in intrahepatic cholangiocarcinoma that can regulate proliferation, and that it sensitizes these tumor cells to palbociclib, and therefore it could be a reasonable marker for those people who would be sensitive to palbociclib, and that's where the diagnostics comes into play, and that palbociclib could be used with compounds from this drug repurposing screen to treat patients with cholangiocarcinoma. And I'm just going to leave it there. I just would like to acknowledge all the people in the lab who did the work. Phil and Kai Ying, who were my students. Sorry, Phil was my postdoc. Kai Ying, who was a student. Danielle, who did the xenograft experiments. And Nikki and Gagan, who were master students in the lab. And of course, Kevin, who is a collaborator at the University of Nottingham. Thank you very much.
So as Dr. Tullo said, we're going to be taking questions at a session at the end. So I would ask you to think up some really interesting questions for both of these two. So our next speaker is Rupert Ecker, who I think is joining us online. Rupert? Yep. Who's going to tell us about precision microscopy and tissue cytometry in cancer research.
Okay, thank you very much. Good morning, ladies and gentlemen. It's my great pleasure and honor to be a speaker in this morning session in this exciting conference. And we are also have been a partner with Professor Dlamini and other members of the research group there at PACRI for several years now. And I would like to give you an overview more from a technical point of view on applications of tissue cytometry in cancer research. When I do this, I wear kind of two hats. One is that I'm an entrepreneur, co-founder and CEO of a medtech company, TissueGnostics, which has its corporate headquarters in Vienna in Austria. And the second hat is that I hold an honorary position as adjunct professor at Queensland University of Technology in Brisbane, Australia. This is where I'm currently. And this is kind of the academic pendant to my research work in TissueGnostics. What is tissue cytometry? I'm sure all of you are familiar with the term flow cytometry. Flow cytometry has been available for some 50 years. It is a very well-established technology to quantify antibody-mediated staining on single cells with one major limitation. The cells have to be in suspension. That's why it's called flow cytometry. When you look in PubMed and search for the term flow cytometry or FACS, FSCS, fluorescence activated cell sorter, you will literally find hundreds of thousands of publications over the past 50 years. With tissue cytometry or tissue image cytometry, we bring this idea of flow cytometry to the slide and to the tissue section and can automatically identify individual cells in tissue context and quantify antibody-mediated staining. Also, multicellular structures can be identified, and something that is called immunophenotyping or contextual cytometry, it's also known as spatial biology, can also be achieved. We can also look inside single cells and look on subcellular compartments, cytoskeletal elements, mitochondria, RNA, and also determine cellular pathogens like viruses, bacteria, or intracellular parasites. Now, why is measurement and quantitative analysis of microscopic images so important? I am sure that all of us are familiar with this famous proverb, seeing is believing, but I would like to challenge this statement a little bit and question whether we shall really believe everything that we see. And let me elaborate on this with the following slide. We see here an artificial images that contains different frames with different shades of gray. In microscopy, especially fluorescence microscopy, we use digital cameras that consist of pixels, and these detect staining intensities in form of gray values. Gray value 0 will be black, gray value 255 will be white. And if we look here on these different frames, then the question to you is, what would you think, what's the difference in intensity between A and B? It is very difficult for the human eye to distinguish, say, 37 shades of gray. That's why I have prepared some answers for you. Answer number 1 means there's no difference. A and B, the gray shades here of these two frames are identical. Answer number 2, the difference is smaller equal 20 gray values in the range of 0 to 255. 3 is smaller equal 50, 4 smaller equal 100, or 5 greater than 100. I can tell you that most people believe that answer 3 or 4 is correct. Now in the next slide, I show you A and B next to each other. In fact, there is no difference. A and B are identical. This is not a trick. It is what is called optical illusion. When we compare A with B in this direction, we can see a difference, and I can tell you also I see a difference. Also, I know that it does not exist. But when we compare A and B here, there's no difference. The reason is that frame A is surrounded by brighter areas, while frame B is surrounded by darker areas. And by looking at it, our brain, which is a very powerful interpolator, starts working and shows us something that actually does not exist. We see a difference between A and B, although it's not there. Now let's imagine we would look here at cells in a tissue section that are stained maybe for a tumor marker, a cytokine, or whatever you might be interested in. Then we would see here a cell in a high background environment and we'd conclude it's tumor marker negative, while here we would see a cell in a low background environment and conclude it's tumor marker positive. Whatever it is, the take-home message is that what we perceive here is not determined by the structures of interest themselves, but actually by their microenvironment. So seeing is not always, or should not always be, believing. And this is a reason why companies like Tissue-Gnostics and solution providers like my company are important and can offer a real support to researchers working with tissue sections and microscopic images. I'm very happy to announce that Tissue-Gnostics will actually celebrate its 20th anniversary this year. The company was founded in April 2003. Our headquarters is still in Vienna in this nice building, very close to the city center, but today we have an international structure of subsidiary and offices on all continents except Antarctica, of course. And by end of last year, our list of reference publications reached or actually exceeded the number of 2,300 reference papers that were published from research groups in more than 60 countries. It's also important to note that Tissue-Gnostics is not only a software company, but we are an ISO certified manufacturer of medical devices. From the very beginning, collaborative research projects have been an enabling factor in the success of my company. And we had more than a dozen national Austrian research projects and also several European ones. I would also only like to mention a few. The very first one actually is linked to a colleague from University of Nottingham, Professor Mohamed Elias, who is the initiator of this 8-Path project. This is all of these projects here are what is called Marie Curie Innovative Training Networks, where we focused on what is now called next generation digital pathology. So bringing analysis into pathology, not only slide scanning, but also digital analysis. Then headed by Medical University of Vienna was the calcium sensing receptor biomedicine project. By University of Cambridge in UK, the anaplastic lymphoma kinase project, translational oncology. Technical University of Aachen was the coordinator of the INSEM project, which looked on cell migration studies. And Helical, headed by Trinity College in Dublin, deals with big data analysis for clinical benefit by using machine learning solutions. And the common denominator in all those projects is that Tissuegnostics is a supplier of high-end solutions for image cytometry, be it classical or machine learning or deep learning based. Just end of last year, we were invited to join another European-funded doctoral network headed by the Erasmus Medical Center in Rotterdam, where they deal with the increasing problem of multi-drug resistance in bacteria. With a university clinic of dentistry and the Danube Private University, we launched a project on deep learning for oral dysplasias middle of last year. And here in Australia, Tissuegnostics is a member of the Australian Research Council funded Center for Cell and Tissue Engineering Technologies, which is jointly led by Monash University in Melbourne and Queensland University of Technology in Brisbane. In my role as adjunct professor, I'm currently building a group that works on or looks on AI solutions for decision support systems for histopathology. And also in Tissuegnostics, we have a similar project where we work on deep learning solutions and including transfer learning, which means that we can move machine learning models from one disease like prostate cancer to another disease without starting the learning process from the very beginning. And we have an international group of collaborators on this project, including team members from PACRI in South Africa. Very briefly, the sub-projects of this research endeavor focus on prostate cancer as a model disease, which we use to elaborate on. Then we want to integrate also genetic probes, which goes a little bit into the direction of single-cell genomics in combination with phenotypic markers, so antibody-mediated staining. The core technology is machine learning and data mining based on machine learning algorithms that automatically finds correlations between molecular markers in multiplexing assays. So when we have dozens of markers stained on the same tissue section, it becomes impossible for the human observer to identify which marker is correlated with which other. And so we use this type of chord diagrams where the lines, where they go, the color they have, the thickness, indicate which marker, and the markers are shown here, like on a clock, which marker is correlated with which other marker. And there's also an academic aspect to this research project, and this is better understanding the black box of deep neural networks. Because in deep learning, we define the input layer, we have a deep learning model, and we see what comes out, but we do not really understand why comes out, what comes out, and what really happens in between. And this is something we are looking here at QUT. So finally, if you're interested in joining this consortium, please contact me either on my Tissuegnostics email address or my QUT email address. We will be most happy to expand our network. Now, a few words about the company Tissuegnostics, its products and solutions. The microscopic equivalent to flow cytometry is called TissueFax. It is a high-end microscope with a scanning stage for 8 slides or a slide loader for 120 slides in the highest throughput version. It works for bright field, fluorescence, with a spinning disc confocal here. Also you can do whole slide confocal imaging, or with a multispectral camera, you can do what is called multispectral imaging of 8 or more markers simultaneously stained, which is independent of the previously mentioned multiplexing. In multiplexing, you restain the same sample and subsequently merge the data set. In multispectral imaging, you acquire up to 8 or maybe even more markers on the same tissue section and do a spectral unmixing. The immediate output of our slide scanners, of our TissueFax instruments, like with any other slide scanner on the market, is a virtual slide. We see here an example of a prostate. In orange is the AMACRA tumor marker, while we have here secretory epithelial cells and basal epithelial cells stained in green. And we can literally see how the tumor here invades the normal area, the normal tissue. This is the green structure here, by the way, is a prostate stone. And like with Google Earth, we can navigate through the sample and zoom in and see cellular and even subcellular structures. I mentioned that we also support whole slide imaging using a spinning disc confocal. We see here a section through an entire mouse brain, and when we zoom in into this region, then we see here a confocal, the details of this, what is called the third ventricle in the brain. What makes TissueGnostics is not the scanner, but the analysis software that comes subsequently. So these beautiful virtual slides are for us an intermediary result. We have 3 applications for the analysis. Most of the data I'm showing today are based on our high-end solution StrataQuest, which is the one for what we call contextual tissue cytometry. Others call it spatial biology. A basic step in any application is that we use the nuclear marker to identify the single cells. And this is a very basic staining of in immunohistochemistry in colon, the brown marker, but using peroxidase DAB is Ki67, a proliferation marker. And the obvious question is how many cells are proliferating? In other words, how many of the blue nuclei also stained in brown? We made a survey among 200 scientists and asked them, what do you think? How many cells are brown positive in percent? And the answers that we received, believe it or not, ranged from 1% to 40%. And I think this shows very clearly that there is a need for quantification, because 1 and 40 is a substantial difference. By doing the analysis in our TissueFax platform, the software identifies the individual nuclei automatically and then quantifies how much brown and blue is associated with each of the cells, and shows this like in flow cytometry in such a scattergram or dot plot, where each dot that you see here represents a single cell in the tissue section. This line here, the cutoff, is very important and well known from flow cytometry. It is set based on an isotype matched negative control that determines the specificity of our staining. And only cells with a mean intensity of the brown of greater than 20 here will be considered positive, and this gives us 14.5%. The important point is that you can do this analysis on Monday morning, on Wednesday noon, on Friday afternoon, in South Africa, in Australia, in Europe, and you will always get the same measurement rather than a more or less arbitrary number. This was an easy example of a marker expressed in the nucleus. Ki67 is a nuclear marker. If we look on other markers that are expressed in the cytoplasm, we use a slightly different approach. We still use the nuclear marker as a master channel, but then generate a secondary measure mask around each nucleus to measure here those cytoplasmic expression patterns. This is a marker called COP1 in epidermis, so it's from skin cancer research. And we see that this marker is expressed constitutively here in the epidermis, but after UV irradiation, the expression increases in 2 ways. The expression level per cell is higher, but also more cells get positive. Normally, people will look at this and say, this is 1+ positive, this is 2+ positive, and your colleague maybe says it's 3+ positive. With our system, you get an observer independent measurement here before UV, after UV radiation, 16.8 before, 44.7 below. But what you also see is this shift of the entire population towards higher values here. So what we visually perceive as brighter. The insert, the quadrant statistic, shows you for the upper left, upper right, lower left, lower right quadrant, how many cells are where, in which quadrant, how many events do we have in total, the number of cells per square millimeter, and the relative percentage. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells between the virtual slide and data space. So you can click on any cell and it starts blinking in the scattergram. Following our motto from image to data, we consider this a step forward and thus call it the forward connection. It also works from the scatter plot back to the image. We can make a gate here, and then the content of the gate, so all the cells which are in this gate, will be highlighted here in red. That means we have an immediate link between a cellular phenotype and histology. Where in the tissue are those cells? Machine learning and deep learning solutions are an important part of our platform. Don't have the time to go into detail, but roughly if you have an organ like this one, or a tissue section like this one, you can label here the different zones in the training phase. This just takes a few minutes, and then the classifier will give you the result of the individual classes you're looking for, and then you can combine this with the single cell analysis. And this allows us to do what we call spatial phenotyping or spatial biology. This is an example of a colorectal cancer that we have received from University of Western Australia. In red, we see CD4, and in green, FOXP3. We identify all the nuclei, the tumor area using the cytokeratin staining here in orange, and then combine these masks and can ask questions like how many CD4 positive cells are there, how many are in the tumor, inside the tumor, how many are in the tumor stroma, and if they're in the stroma, how far are they away. We can obtain such a scattergram where each dot that you see here represents a CD4 positive T lymphocyte. So only CD4 positive cells are shown here. On the x-axis is the size of the nucleus of all the cells, and on the y-axis is the distance in micrometer from the next tumor area. So we can make these gates here, 1 to 25, 25 to 50, 50 to 100 micrometer, and so on. And then use the backward connection and visualize here in red all those cells, CD4 positive cells, which are just outside the tumor, but not further away than 25 micrometer. Go here, those which are in a distance of 25 to 50, and so on. If we go back, we see here in this quadrant statistic that in total, we have 7,234 CD4 positive cells. 2,267 thereof are in a distance of 1 to 25 micrometer. This is 31%, 22% are in the distance zone of 25 to 50, and so on. So we see there's a clear gradient away from the tumor. And with this, I would like to conclude my presentation. I hope I could show you that with TissueFax, the microscopic equivalent to flow cytometry, we can support researchers to move from image to data and thereby increase the observer independence and reproducibility. I would like to point your attention to a dossier on artificial intelligence in biomedicine that has been published last year in collaboration with our partners around the world, and one of them actually being Professor Zsolt Dlamini, another one being Professor Stefan Barth, both from South Africa, and also other researchers around the globe have contributed to provide a global outlook on the topic. I would like to sincerely acknowledge the contribution of our academic partners in Europe and across the world, like from the different EU projects that we participated. Also my local partners here in Australia, Jyotsna Batra from Translational Research Institute in Australia, Professor Bida and Professor Dlamini from South Africa. And last but not least, I would like to emphasize that our local business partner, Margie Hengsberger, also joins the conference and is present with a booth just outside the lecture hall where you are. She runs a company that is called MIT, Measuring Instruments Technology, and as such, she represents Tissue-Gnostics Africa division. And she will be happy to talk to you and have any further discussions. With this, I would like to conclude my presentation. I hope that I could show you that with TissueFax, you can analyze and explore cells, the fundamental unit of life, with a precision that inspires. Thank you very much for your attention and enjoy the conference.
Thank you very much, Rupert. If you could just stay online while we take some questions. Could I ask Sheila and Kevin to come up to the front here? No, you can't hide. And I'd like to just throw this open for questions for a few minutes. While you're thinking of them, I'm going to start. This is sort of to both of you, because it's clear that in prostate and cholangio, there is a difference in the effect of PRH. Is that difference because of the post-translational processing? In other words, does PRH get cleaved in prostate, but not in cholangio and colorectal? And that's why in cholangio and colorectal, phosphorylated PRH is able to activate transcription, and in prostate, it's not.
It's something that we would definitely like to look into. It's a possibility that it could be to do with the post-translational modification, and it's something that we'd like to investigate. But it could easily be to do with cofactors that are being expressed in one lineage rather than another. It could be something to do with the opening of the chromatin, the availability of chromatin in one lineage than another. So there are multiple possibilities, really.
So do you know if proteasome inhibitors have an effect?
We haven't yet looked at that.
Because it could be that in myeloma, when people are given proteasome inhibitors, it changes the behavior of the PRH.
It could do, yeah, absolutely.
Do you have any other questions that are coming up? Yep.
As a clinician, I always think about targetable moieties, and the ability to target transcription factors has always been very problematic, but the upstream, the CK2, seems to be readily targetable. And I was just sort of wondering about a brief synopsis about CK2 inhibition and its cancer role. We had a brief discussion last night, but I thought it'd be beneficial for the whole group.
I think CK2 is very targetable. There are lots of different inhibitors. The trouble is, it's like all the kinase inhibitors that Dave knows a lot about. They're not all as specific as they need to be. So, for example, I showed you some data there with CX4945, but that does inhibit another kinase. And so it really depends on what kinases are expressed in the cell that you're targeting. So you can throw the same kinase CK2 inhibitor on two different cell types and have very different effects, because you're not inhibiting the kinase that you think you are. So, but having said that, if you have a tissue where there's a very high expression of CK2 and maybe the other kinases aren't there, then you can certainly use that in inhibition. And we had a paper with Sheila, I think last year, where we were using CK2 inhibitors in combination with cisplatin, and you could then see combinatorial effects depending on, because you're inhibiting, if you inhibit CK2, you're inhibiting a whole bunch of things like DNA repair, you're inhibiting cell migration, cell proliferation, lots of different, that's another problem, you're inhibiting lots of different things. But having said that, there's a lot of clinical trials going on at present where CK2 inhibitors are being trialed as combination therapies. And that's one of the things that we're hoping to investigate more, is the combination of CK2 inhibitor with other inhibitors, or with immunotherapies as well. There's quite a few trials looking at immunotherapies supplemented by CK2 inhibition. Not that we're doing that, but that those are going on.
Yeah, thank you very much. I'm Tiperi Luengo, the surgeon from VETS. The PRH in prostate cancer was an early event, as you presented, that it tends to be marked in early on, and as the cancer progresses, it gets lost. And is it the same setup in cholangiocarcinoma? Is it influenced by the stage of the disease? Thank you.
I don't think we've yet got to that ability to get cholangio in very low grade. I mean, just getting samples is really quite difficult. We have access to cholangiocarcinoma samples, but they're not graded that easily. They're mostly very advanced cancers that we have access to. But what we do want to do is look in primary sclerosing cholangitis, where we can see maybe the transition from the cholangitis, the inflammatory condition, towards the cholangiocarcinoma condition. And I think there we might be able to tell a bit better. But no is the answer. We haven't seen that kind of change and progression. But to be honest, we haven't really started to look.
If I can just ask a quick question to Rupert. What is the likelihood of being able to expand the multiplexing or the multiple fluorescent markers to what we're starting to see with things like NanoString, where you can use hundreds rather than fives or tens of markers using different imaging technologies?
So we are working on solutions where you can use dozens of markers. There are existing technologies like MELC, for example, multi-epitope ligand cartography, where they have demonstrated up to 200 markers, antibody-based. But frankly speaking, we are not at the level where those genomic companies are with thousands of markers per cell. Although we are in discussion and negotiation with a few of those providers to combine the analysis platforms.
Question. I wanted to ask a question about that as well, which was, we've used the NanoString technology and their software doesn't seem anywhere like as good as your software. So is it possible to use, mix and match between, say, for example, their data and your software, and vice versa?
Mm-hmm. This is actually what I meant, what we're currently negotiating, to integrate the data from NanoString or 10x Genomics or Merfish and combine them with the format that we have, with the antibody-mediated analysis. But this is in progress.
Question. Hi, my name is Dolly from the University of Limpopo. My question is for Professor Kevin. You mentioned in your study that you guys were looking at PRH responses in UK samples. So I just wanted to know if there were any regional specific differences in UK samples versus Kenyan samples, which I saw you said that you guys are developing a bank for. Like you guys are doing the same research in the UK as you're doing in Kenya on the same, on PRH.
That's a very good question. That is why we're doing our current study. I wish I had more data to show from that study, but unfortunately, because of the pandemic, we just don't have it. But the logic for it, the rationale, is that we were interested in why in Africa prostate cancer seems to occur, certainly in Kenya anyway, that I know about. Prostate cancer seems to occur in much younger individuals and to be a much more aggressive disease than it is in the UK. So in the UK, we're very interested in, for example, are there differences between the white population and between black people in the UK? But we don't have enough patient groups, we don't have enough patients from, say, for example, from a black heritage to, or an African heritage to make good comparisons. So by going to Kenya and working with our Kenyan colleagues, we're hoping that will tell us more about regional differences that in the UK it's very difficult to measure, and we haven't even tried because the patient numbers that we can look at are too small.
I think if there are any other questions, we'll have to catch the speakers during the tea break. We've gone quite over time. So we'll be going on to our tea break now. I'd like to thank the speakers and the audience for the questions and for attending, and we'll meet again at 11.
Thank you very much. Enjoy the conference.
Morning, everyone. I'm Dr. Mbata, chairing the next session. I'm the surgeon from University of Pretoria with Dr. Hal, who's our research manager at PACRI. In the interest of time, we're already 30 minutes behind. We will just move into the next session. The theme of this session is precision health equity, which is an interesting topic for us, particularly in South Africa as a low middle income country, and also a country that's very culturally diverse with multiple people coming from diverse cultural groups. So our talks will look at some of the practical ways we can use to achieve equity in precision medicine. So the first speaker is Professor Melissa Davies. She's ready. Okay. Professor Melissa Davies, who's a scientific director of the International Center for the Study of Breast Cancer Subtypes and director of health equity at England Institute of Precision Medicine, an associate professor of cell and development biology in the Department of Surgery at the Weill Cornell Medicine in New York. She's holding an adjunct faculty appointment in the Department of Population Health Sciences at Henry Ford Health Systems, and she obtained a PhD in molecular genetics at the University of Georgia, Athens in the USA. Professor Davies.
First, thank you for the invitation. I'm so happy to be here in South Africa. I've been in touch with Dr. Dlamini for a couple of years now, looking forward to establishing some more concrete collaborations. So today I'm going to talk to you about some of the work that we've been doing over the past, over a decade now, trying to resolve what is causing disparities in breast cancer. So the title on the slide is a little different than the one in the book, but trust me, it's pretty much the same. But I want to touch upon some of our more recent findings in terms of tumor immunological processes. Here's my disclosures, and also I'll just mention that I'm also newly appointed as the inaugural director of the Institute of Translational Genomic Medicine at Morehouse School of Medicine. So that's a relatively new role that didn't necessarily make the program this time. So just to start out, to talk about what I consider the elephant in the room. So currently, especially in the United States and in any sort of healthcare system where we have a lot of marginalization of minoritized populations, there's a bit of debate about what causes the disparities in clinical outcomes. Of course, we know that there are surely social determinants that can impact health generally, but we also tend to see on a population level differences in biology, so the plasticity or the epistasis of certain diseases. And all of this can be linked back to molecular genetics. Particularly on the population level, this would be linked to genetic ancestry. And so what I'm going to hopefully show you today is that we actually need to combine our efforts on both strata in order to reach what we consider health justice. Particularly, recently I was asked to give sort of my predictions of what would be some of the highlights of cancer disparities research in the coming year. And this kind of just gives a broad overview. Particularly today, what I'm going to talk about is the one here in red called oncologic anthropology, which is a term that was coined by our research partner at Weill Cornell, who's the chief of breast surgery, Dr. Lisa Newman. And then also we're going to talk a bit about ancestry-related genomic diversity across populations. But mainly for that project, you'll hear much more about it later after lunch by one of my colleagues, Nicholas Rubin from the New York Genome Center. So this is just an outline. In the interest of time, I'm going to just kind of skip over that. But this is the problem, that breast cancer disparities in the United States, the breast cancer mortality rate is more than 40% higher for African American women than our US Caucasian or white American counterparts. But this wasn't always the case. So what these graphs basically show you on the left are the mortality rates for both prostate and breast cancer. Now the map on the right, I believe it's your right, yes, actually shows the concentration or density of poverty county by county across the United States. Now, later on, I might show you a map that also shows you the density of the minoritized Black population in the United States. And what you would see is that it overlaps, because traditionally minoritized populations tend to be at the lower SES or socioeconomic end of the spectrum. And so we know geographically, if we look at where our minoritized populations live, this would also be regions of our most impoverished populations. But poverty is not exclusive to minoritized populations. So on the graphs on the left, what I'm showing is not only the rates of mortality for Black and white populations, but in more recent analyses of our surveillance of US death rates for prostate and breast cancer, these Black and white populations have also been stratified for whether or not the patients lived in poor or affluent counties. So patients who live in affluent counties, therefore, are not necessarily burdened with what we would consider a lack of access or lack of ability to receive curative therapies. So if socioeconomic status or poverty were really the only reason that we would see the disparity, then we should see the resolution of both of the affluent populations having pretty much the same mortality rate. But instead, what we see is that our Black population, even living in an affluent county, still has a higher mortality rate than the white population living in poverty. And so that tells us that some of this actually still transcends socioeconomic status. In the case of breast cancer, what these arrows are actually pointing to is sort of the dissection of the trend, which is that prior to the early 1990s, it didn't matter if you were Black or white, we all had the same opportunity to survive breast cancer. The disparity actually started to emerge at the onset of targeted therapies that were mainly designed for endocrine signaling in breast tumors. And at that point, we see this stark decline in mortality only in the white population. And this didn't necessarily start to translate into the Black population until nearly a decade later, but then we still see this persistent 30 to 40% mortality difference. And so sort of when I started my trek into disparities of breast cancer was during my postdoc work at the University of Chicago, where I worked with Dr. Fumio Lopate and Dr. Sarah Gellert, who were the co-leaders of the Interdisciplinary Cancer Disparities Research Institute. At that time, one of the things we wanted to start honing in on is whether or not there were distinct biological differences between race groups. But the first thing we wanted to know is whether or not we could detect that same disparity that was seen on a national level. And so at the 5-year survival line, we do see sort of this disparity between African Americans and Caucasians in terms of their survival, and this is looking at all types of breast cancer. But then when we stratified for ER positive versus ER negative tumors, in the ER positive category, that disparity went away, primarily because the treatment was effective in both populations. Again, this was from the same healthcare system. So everyone had the same standard of care. But then when we looked at the ER negative tumors, we actually saw an even broader disparity at the 5-year mark, which indicated that the treatment wasn't necessarily equitable for both white and Black patients. So now we think about then the tumor biology that sort of is the landscape of how we determine how patients are treated. We know that from the best to the worst prognosis, there are different molecular markers that can be used to define different subsets of breast cancer tumor types. On the worst end of that spectrum is triple negative breast cancer, ER, PR, HER2 negative, which happens to be more frequently diagnosed in African American women, and it also tends to be an earlier onset disease. Now that earlier onset also gives us an indication that there's a genetic predisposition. So now we want to know, what are the clinical differences? You know, we come back to this case of, can screening save us? Of course, we know, especially in the case of triple negative breast cancer, where we don't have targeted therapies, the earlier the diagnosis, then the better off the patient will be. It's easier to cure an early stage disease than a late stage disease. And so in this study, we looked specifically at our Henry Ford Health Systems cohort, as well as the New York Presbyterian cohort. These are all patients who were diagnosed with triple negative breast cancer. It's about 20 years worth of data, almost 800 patients. Advancing, okay. So the first thing that we note is that there was not a statistically significant difference in our white versus African American population in terms of whether or not their tumors were screen detected. So whether or not a tumor is screen detected is really simply what it sounds like, that if it was diagnosed because of mammography, typically the patient doesn't even know there's a mass, which means it's usually very small and it's an early stage disease. If it's non-screen detected, then that means something sort of compelled the patient to come into clinic, typically some type of pain, or she herself identified a mass. However, we don't see differences in stages, at least not significant differences between our white and Black population. So now if we look at the survival rates or probability of survival, if we stratify for non-screen detected and screen detected, we see what we expect, which is that the earlier stage screen detected cancers tend to have the best overall survival. However, when we stratify now for whether that same trend exists in white and Black populations, again, we see this larger gap in survival for non-screen detected cancers in African Americans. And again, this points to the fact that the treatment does not necessarily behave the same in these patients. So about, I don't even know how long this was ago. I don't want to tell you the date is here, but I don't want to date myself. I'm getting a little older. But what we wanted to know is whether or not we could detect differences in the tumor biology of triple negative breast cancer. So the issue with the triple negative diagnosis is that it's a diagnosis of exclusion, meaning all it really tells us is that there are no targets that we can currently use to treat. So it's this sort of catchment, if you will, of everything else. And so one of the things we identified was that African Americans also in that triple negative category were more likely to have a quadruple negative subtype, that fourth hormone receptor being androgen receptor. In that same study, we went on to show that quadruple negative breast cancer was even earlier onset than triple negative breast cancer, particularly in Black patients. Again, another clue that this is a genetic predisposition. In later studies, we looked more carefully at the gene signaling, and unfortunately, at this point in time, we did not have the tools necessary to do more than stratify by self-reported race. Okay, so these are categorical strata that patients basically self-proclaim that they're African American or white American, but we were still able to see these stark contrasts in some of the molecular biomarkers. This last study was actually microRNAs, where we saw so many differences in a subset of microRNAs. We could even use those microRNAs, the expression of them in tumors, to predict race. We saw that some of them were regulating some of the canonical cancer pathways, such as MYC. And then we could detect even statistically significant differences between the quadruple and triple negative subtypes. So this is, again, another way of defining a new subtype of breast cancer and defining molecular markers that could potentially be targetable pathways. The issue that we run into here is that the clinical significance only exists in the African American population. So if you look at those survival curves, you'll see, for instance, that, for instance, one of these markers, MIR17, only had a clinical difference for African Americans. And so here's where equity comes into play, right? So most of these studies, we wouldn't have been able to publish in higher end journals because most of the journals would have, kind of seen these as smaller populations, and it's not largely applicable, right? It's not readily in interest of the broader community. But what we want to basically say and sort of move beyond those barriers is that even if we look at every definition of a subtype of breast cancer, we do see population differences in simply the incidence or distribution of those different biological subtypes. And the differences in those biological subtypes, again, could be the difference in how and why we can treat these patients more effectively. And so if there is a genetic driver of triple negative breast cancer, classical methodologies would be able to unwind or unravel what they are. Particularly, this is a pedigree that was used by Marie Claire King and others when they discovered the BRCA gene. And so the first thing you should note is that these are very large families with very well annotated health history, going back 3, 4, 5 generations. And this does not exist currently in our African American population due to our social history in the country. And we can discuss that later. But then GWAS studies should have saved us from not having sort of this well-annotated pedigree analysis by virtue of interrogating hundreds of thousands of individuals and across millions of different genotypes. We should have been able to identify then what is the genetic predisposition that's driving triple-negative breast cancer. But what we have here is a sampling bias problem because the majority of the patients that were used in GWAS studies were of European descent. And here's the data that represents that. So over 98% of the GWAS data that we currently have is generated on patients who are of European ancestry. Despite this group not being the world majority, the largest population on the planet actually is represented by less than 1% of the data. This translates into clinic by having an accurate clinical tool. So the bar graphs on the right actually show the rate or likelihood of having what we call a variant of unknown significance if a patient who is African American, Hispanic, Asian, Middle Eastern are given these mutation panels. What's going to happen is we may find a mutation in their gene, but 40 to 45% of the time it will be a variant we've never seen before in a GWAS study. And so then by definition, it's unactionable because it's a variant of unknown significance. So what we take is an oncological approach, a global approach, where for instance, for triple negative breast cancer, we assessed what was the rate or frequency of triple negative. And what we found was that the highest rates were within the sub-Saharan African region globally. This is the region that has the highest rate of triple negative breast cancer. And then on the continent, the West African populations have the highest rate. Interestingly, other countries that we can stratify for self-reported ancestry, so African Americans in the US, Black South Africans, for instance, also have the highest frequency of triple negative breast cancer. And we see that also repeated in other countries, including the UK. And so now we think about the African diaspora. So how will we be able to hone in on an allele that could potentially have a unique signature or that drives a unique biology or a unique predisposition? In order to do this, we have to consider the fact that the African diaspora is unlike any other human population by virtue of typically the populations that we know of today, how we define them with ancestral informative markers were due to strategic migration, okay? So these would be founder populations where a single event moved a group of people from one region to another. In the case of the African diaspora, what we have is the result of the transatlantic slave trade and the East Indian slave trade, where we have this continuous gene flow over centuries. Millions of Africans were displaced. And so now this is why we think we can see sort of this distribution of triple negative breast cancer within every country and continent that we look in, having a higher rate of triple negative breast cancer. So our ICS-BCS cohort is a multi-ethnic cohort where we have actually been collecting samples by recruiting patients from across that spectrum of the diaspora. So we have several sites in Africa, in the Caribbean, and then multiple sites in the US. And one of the first things that we did with these data is measure West African ancestry. And what we found was that the patients who had the highest component of West African ancestry were more likely to have a triple negative breast cancer diagnosis. We even saw that if we only looked at our admixed African Americans, that the component of the population of African Americans who have the highest rate of West African ancestry also were more likely to have triple negative breast cancer. And so now here's just a snapshot of some things that are in progress as we now look from a whole genome sequencing perspective. So now not just measuring genetic ancestry, but also measuring regional ancestry for each one of these patients. And we're looking at things like mutational signatures across the spectrum, also homologous recombination deficiencies. And what we're hoping to do is hone in on what are the causality drivers of either homologous recombination deficiency in these populations, but most certainly unique mutational patterns that tend to be more correlated with African ancestry. So these are the cosmic the cosmic mutational signatures. And what we tend to see is that in our population of African patients, we're seeing a different sort of spectrum of some of these mutational signatures. And so what we hope to do is hone in on what some of those molecular drivers are, particularly for homologous recombination deficiency, because we know that there are targeted therapies that are indicated by homologous recombination signatures in the tumors. Interestingly, one of the things we see, which is linked to signature 3 in our African ancestry patients, is sort of this trimodal distribution where we see either high rates of signature 3 or sort of moderate rates of signature 3 or no signature 3 at all. And so what we think is happening is not only in this homologous recombination deficiency context, but we believe this is sort of the distinction of a unique DNA repair gene mechanism that we're starting to investigate. And hopefully, if invited back next year, I'll be able to show you more data. But in the interest of time, I wanted to give a synopsis of our cancer discovery paper, which looked at RNA-seq of triple-negative breast cancer tumors. And one of the more unique aspects of this analysis was that instead of using self-reported race, we quantified genetic ancestry, and instead of then putting people back into boxes based on an arbitrary threshold of their ancestry, we used it as a continuous variable for a linear model to establish whether or not there were any gene expression or networks that were associated with African ancestry. And so we found what we were looking for. There are definitely genes that are associated with African ancestry, meaning there is most likely an EQTL that is African specific that are driving the gene expression networks. We also found genes that were European centric, though unapologetically we focused on the African networks. And then in this blue bubble at the bottom, we still found some genes that were strongly associated with self-reported race and not associated with ancestry, and I'll come back to those. So this is sort of the spectrum of genetic ancestry for the cohort that was used in this paper. The spectrum of gold or orange are the African ancestry, greens are European, blues are South Asians, looks like, and purples are Native American. And so we see what we expected, which is that our African American cohorts, and I don't know if my laser pointer works very well here, but in our African American cohort, we see the spectrum of genetic admixture primarily from Europe. But when we looked at regional ancestry, what we found is that in every African American, there is every region of Africa represented in their genetic background. And not only that, but where or the origin of their European admixture tends to be very broadly disparate. This tells us that the genetic makeup of this group of individuals is very heterogeneous. And so then putting them into one group is problematic when we're trying to identify a genetic predisposition. And so then the first step when you're looking for a genetic causality would be to measure and quantify the genetic background. And now what we're doing as a follow-up to this study is looking at localized ancestry across the genome to help explain some of the findings that we saw in this paper. Primarily, one of the biggest things that we saw is, and we've seen this before in other types of studies, is that West African ancestry was associated with a higher infiltrate of immune cells. Now, we did this using a deconvolution algorithm called CyberSort. And so we saw overall there was a higher immune cell representation in the tumors, in the triple negative tumors of people of higher African ancestry. But more specifically, there were specific types of immune cells that were more present. We validated this using IHC for an independent cohort across the spectrum of our ICS-BCS patients. But interestingly, when we think about sort of the immune signature in tumors, typically we would think that having a high amount of immune activity would lead to better overall survival, but that's not what we actually see in African Americans. So here what I'm showing are Kaplan-Meier curves based on Tau scores, where we see that higher infiltration in European Americans leads to better overall survival, but we don't see that benefit in African Americans. And partially what we think this has to do with is the status of the immune cells that are infiltrating. And so in that paper, we actually did see that some of the more immune suppressive markers were more highly correlated with West African ancestry. This isn't necessarily a new concept. We know that if we were to take immune cells from people of African descent and European descent and challenge them with the same, for instance, bacterial pathogens, that they would have completely different transcriptional responses. This is partially because of natural selection, some of the selective pressures that are in, for instance, sub-Saharan Africa. And one of the genes of interest right now is the gene called DARK, or ACKR1, where there is an ancestral informative marker called a Duffy null allele. Now, this mutation actually removes expression of the protein only on red blood cells, and the function on red blood cells is to regulate systemic inflammation. Its expression on endothelial cells is to help sequester, or I should say, recruit immune cells into interstitial tissue. But when we look at the distribution of the Duffy null allele, we see that it actually tracks with, for instance, the result of the transatlantic slave trade. Now, we actually showed that in breast cancer, expression of this gene corresponds with higher infiltration of immune cells, which corresponds with this function on endothelial cells, but we also see higher inflammation in tumors that have high expression of DARK. In that first study was in TCGA data. We've gone on to replicate those findings in our ICS-BCS cohort. But more importantly, now we're using more advanced technology to look at the immuno landscape on a spatial spectrum, where we're seeing, for instance, high DARK tumors versus low DARK tumors having very distinct patterns of infiltration. So on the right, what we're showing is a DARK high tumor that has much more infiltration of immune cells that corresponds with the CD31 marker, which is the endothelial marker in those tumors. More importantly, when we look at the status of those immune cells, we see, for instance, when we isolate the single cell analysis, that these are actually CD8 positive T cells that were in the tumors that we've been interrogating. This is preliminary data from about 4 cases, 3 regions of interest across the tumor space. In a DARK high environment, we see certainly a much larger population of those T cells, but we also see the signature of exhaustion, where the red actually indicates PD-1 expression on those CD8 positive T cells. When we look specifically at which patients we derive those PD-1 positive T cells, the majority of them came from our African American patients. So this corresponds to what we think is the circulating impact of having a higher inflammation, chronic inflammation. And so now we're actually looking at systemic markers related to the chemokines that the DARK receptor regulates. And to close, I just want to point out that for those self-reported genes, the genes that were associated with self-reported race, we looked more carefully at, and again, these are people who all have African ancestry, but we saw a unique signature that was specific to African Americans. And those particular gene pathways were in comorbidity pathways related to obesity, diabetes, and cardiac metabolic pathways. And so this is how we believe structural racism becomes biology, because we already know from the social sciences that living in a marginalized community, redlining, having overexposure to toxic sites, which is what the map on the upper right is showing, we tend to have a higher rate of obesity and diabetes in this population, and all of that tends to transform the imprint of the gene pathways that are on our tumors. But instead of this being a social issue, it could be a precision oncology issue, because we know that these are the patients that are coming into clinic, and we know that these are on a population level. These are the signatures that their tumors are expressing. And so this is an opportunity to start targeting these pathways in this patient population. And with that, I'll close, acknowledge our funders and all of our ICS-BCS collaborators, New York Genome Center, as well as the Englander Institute of Precision Medicine at Weill Cornell. Thank you for your attention.
Our next speaker is Professor Rui Reis from the, well, he's from both Portugal and Brazil. In Portugal, he's a professor in the in human genetics, at Manoa University, and he's also the leader of oncobiology group at Barretos Hospital, Cancer Hospital in Brazil.
Okay, good morning, everybody. Thank you for the introduction. And it is a great pleasure to be here with all of you, with the distinct guest of speakers. And I want to thank the organizer, Professor Dlamini. And I will present some of the work that we have been doing in Brazilian. The work that I'll show here is from our research institute. And how Ancestry also could link. So I'll present just a little bit of our institution, and then the genomic part of melanomas, and then the epigenetics that we are performing. So first of all, I'll actually speak a little bit of our institution. So Barretos Cancer Hospital is a private non-profit institution, so we attend only 100% free of charge. From the beginning, our core was, our philosophy was to take care of everybody with care and compassion. And we are focused on underserved and remote population areas. So our hospital, as you see, is located in a small city in the state of São Paulo, Barretos City, that is 500 kilometers from the capital. So it's a rural area. And so we attend with this philosophy and free of charge patients, so most of them are poor people. So they come from all over Brazil to be treated at our institution. And since some patients travel more than 3,000 kilometers and with the aim of being more close to the patients, so the hospital now is spread all over the regions, more in less developed regions of Brazil. And currently we have 7 treatment units. From beginning, we were focused on prevention, so we have 24 prevention centers. And since, like in Africa, Brazil is a huge country and the infrastructure is not good, it's difficult for the patients to arrive to the hospital. So we focus also in a strategy of mobile units. So today we have 35 mobile units together with a factory in our own facility that do these mobile units to go around the country. So along with that, so we have approximately, well, we have a lot of, I don't find here the number of cases, but we have around 16,000 new cases of cancer per year. So it's the biggest cancer hospital in Brazil. So the hospital was focused on attending with good quality the patients, and 10 years ago started the research area. So it was focused in translation research, so we want to apply biomarkers to our population, to understand our population. So it has a genomics and core facility. Since many years, we have institutional tumor bank. All patients that arrive there, if they consent and they sign, so we have blood and tumor tissue, saliva, feces, among others. So we are the biggest tumor bank in Latin America with today is more than 3,000 samples. Everything is stored with quality controls. And also we have for functional biology, we want to understand what is biomarkers, but then understand their functions. So cell biology and PDX. So we are also developing primary cultures from our patients, since they must be different from the ones that are commercially available. So we can find in our website some of the highlights of the institution. So as already presented here, so we have a big disparity in genomic data. So if in Africa it just represents 0.1%, in South America it's even worse, the scenario. And in Brazil, with the admixture of population, so the genetic ethnicity is even less understood, and this is even more complicated. So we try to diminish this gap. So in 2013, we started a cancer sequencing initiative. So it's one of the biggest in Brazil. So we are doing a lot of sporadic cases as well as hereditary, so whole genome sequencing. Here, just research. Along with the research, we have a diagnostic lab that follows a clear rule, so it's different. So this is for research, so large sequencing. And I'll present here some of the data on genome, but I'll be pleased to discuss other projects that we are developing. So we started this, so the idea was to perform whole genome sequencing. We'd pair, all of them is pair blood and tumor of 100 patients. So we did all of them validation by target sequencing. This was done in collaboration with Mendellix, and then when the answer we have done a collaboration for bioinformatics. These were part of ICGC. So we participated, so the only Brazilian project on the ICGC, and we participated with melanomas because the other two projects that were available were melanoma from USA and from Australia, and all of them only had cutaneous melanomas that arrived in in lighter skin people. And in Brazil, we have, along with those cutaneous melanomas, acromelanomas that arrive more in darker pigmented skin, and is really poorly understood. So the first result, so as expected, melanomas are the highest mutated tumors due to the exposure to the carcinogenic UV mutation. So together with Australians, our project, our tumors were among the highest mutation per megabasis. As mentioned here, so doing this type of approach, we can have the mutation signature and then can have a hint of the etiological factor. So as expected, most of our cases have the signature 7 that is related to UV exposure, but we have a rather amount of patients that don't have this signature. So we're more associated with this acro subtype. When we compare with the mutation profile that is driving today the treatment, so we have here the most 20 genes that were discovered by TCGA. And currently melanomas are divided in the molecular subtypes. So we have the BRAF subtype. We have more than half of our cases belong to this subtype, the RAS subtype, the NF1 subtype. And we want to identify the triple negative, the ones that don't have mutation on this one, is much higher than was reported. And it was associated with this acro subtype. So as expected, the typical genes that are known for cutaneous melanomas, they are not present in darker skin persons. So here, just a small comparison. So there's several differences, and NRAS was one of the really striking difference. And this can have an impact in the future in target sequencing. Today, we don't have a special drug, but in the future, you can observe that this will be a less attractive target for this type of tumors. We did also another type of analysis, but to the sake of time, I will not present. And one was to integrate the mutation profile with copy number alterations, with structure alterations. And there were several pathways that were deregulated. And one that was striking was methylation and histone modification. And it is known that epigenetic mechanisms that can be surrogate markers of the exposure, of the risk factors that were involved. And UV is one of the major factors, as expected, but for darker skin is less reported. So next, we integrated our genome project with epigenomic analysis, so in collaboration with the International Agency for Research on Cancer. So we divide our study in first part was compare the cutaneous melanomas, the ones that have UV signatures, than the ones that don't have. So if it's even in cutaneous melanoma, the UV signature can drive different pathways. We compare our data with the TCGA data, and then wondering if the acromelanoma that is more present in darker skin persons. So we want to compare our cutaneous with our acro in terms of the epigenetic markers and if there is some difference. So just to give you some of features. So as expected, white skin is more prevalent in cutaneous, both in our cohort and TCGA, and acro in pigmented skin persons. So UV signature, that is the majority of the cutaneous, is really not present, or only 90% of the acromelanomas, and then the molecular subgroups that are already presented. Importantly, these also have a prognostic impact because only in the cutaneous melanomas, so the ones that have a UV signature, they have a better outcome than the ones that don't have it. So this can also have an impact on prognostic of the patients. So we want to understand a little bit more. So we did the methylation profile of the cutaneous melanomas associated with UV and non-UV signature. So we identified two main clusters that were associated, epigenetic clusters associated. So they were heavily related with immunological pathways. Some of them associated with skin, like systemic lupus erythematosus. So we found that are really epigenetically they have two different pathways. And we want to understand which are the genes, the epigenetic markers that most discriminate these both groups. So after a series of filters, so we end up with 3 genes, and one of the genes, TAP-BP, so is the one that was most interesting. So was hypomethylated in UV signature and hypermethylation in non-UV signature. So the signature, the methylation of TAP-BP was associated with the expression profile. The one that was hypermethylated has a less expressed. We also validate these findings by pyrosequencing in our data, so it's not an artifact of the epigenetic analysis. And also importantly, we saw that the UV signature associated with the prognostic, when TAP-BP was also methylation or not, was associated with the prognosis of this patient. So we identified here a marker of potentially of UV signature that is really important in this context. So next, we want to compare the acro with cutaneous melanoma. So this was done only in our cohort. And we showed that cutaneous melanomas, non-UV signature, they are similar to acromelanomas than to cutaneous melanomas that don't have, that are UV related, have a UV signature. So it seems that the etiological factor is more important in the mutation than the subtype of histological subtype of the disease. So we tried to identify with genes. So we found again that so with associated the acro has a worse outcome than the cutaneous, and try to identify also which are the epigenetic markers that distinguish this. We identified several features, including TAP-BP. However, the number of cases were smaller to have a clear statistical significance. So we are cautious about that. But so in the end, so identified a gene signature, epigenetic signature associated with UV exposure, and we are now looking for which are the genes that are more present, either the mutation and epigenetic markers in the acro patients. So to finalize, so I'd like to thank you to our Barretos Cancer Hospital that now changed the name, so Hospital de Amor, Love Hospital. In English is a little bit quirky, but if you go there, you feel that, because everybody really, the first thing is to treat as a person. You take care of a person, then you can take care of their cancer. So I found a little bit weird, but now I incorporate it, and it really is the feeling of the institution. And so everybody, our CEO, and the people that were most involved, and of course our collaborators from MD Anderson, IARC, and Mendelix, and the funding agency that allowed us to perform this work. So thank you very much for your attention.
Thank you. I think we'll take questions afterwards. Maybe we can have the third speaker come in. Our third speaker is Professor Reed. Professor Reed is the president and CEO of HGT Bio, a Seattle-based biotech company developing next-generation RNA technologies for installment and prevention of cancer. He's an adjunct professor of medicine at Cornell University Medical College and research professor of pathobiology at the University of Washington. Professor Reed holds a PhD in microbiology and immunology from the University of Montana. Thank you, Professor Reed.
Thank you for the kind introduction. I'm happy to be here. I'm a scientist that focused my whole career on vaccines for neglected diseases, parasitic diseases, tuberculosis, etc. But along the way, tried to adapt our technologies to oncology as well by creating biotech companies to help develop these. And the latest one is what we call HGT Biotech Bio in Seattle. HGT means host-directed therapy. And as you see from our logo, Immune Therapy for All, we're focusing on really next-generation RNA technologies that can be readily adapted to all kinds of populations around the world. RNA technologies, we've been working on these for about 10 years now, and the first generations have been very clumsy. You may know there are 3 major RNA companies that were founded in the world, and they all met with a lot of failures. In fact, they were all about to probably close except for COVID, which provided a unique opportunity with a defined antigen and relatively easy immune correlate, which is antibody production that was needed to be made rapidly. But the problem with these technologies is they weren't widely available. They're -80 freezing storage conditions, et cetera. So we went about early on to try to identify better ways to make RNA vaccines. And this portion of the talk just talks about messenger RNA versus self-amplifying RNA. And there's a lot of complicated aspects, but really what I'd like to point out is that the messenger RNA technologies readily available today are not made to generate potent T-cell responses. They'd like them to be, but they don't do that. And in fact, there was a New England Journal paper published 2 weeks ago with Pfizer vaccine, actually BioNTech technology, showing that people that were boosted with this vaccine had almost 0 T-cell amplification, 1.2 or 1.3 times increase, so negligible. So mRNA is not potent for developing T cells, the kind of responses that we'd like to have in oncology, CD4 and CD8. Self-amplifying RNA is something that we got excited about many years ago. My last company, I started with David Baltimore, who he won the Nobel Prize for reverse transcriptase discovery in HIV, but David had a lentiviral platform that was very exciting to me because it was a self-amplifying RNA that was very potent for T-cell responses. Problem is, very, very difficult to produce, very, very expensive. So it didn't fit with our theme of immune therapy for all or widely adaptable platform technology, but it showed us the proof of concept that if you have a replicon, a self-amplifying replicon, you can get very potent T-cell responses. And so what we did, and what replicons do, is they amplify the RNA signal within the cell, so they give you more copy numbers. They also form something like a particle. So we went about trying to form a platform technology that could give us the advantages of a lentiviral or an adenoviral vaccine without the disadvantages, which are the manufacturing and the envelope, and in all cases of viral vectors, the problem of pre-existing immunity. Even if you don't neutralize the adeno on the first shot, you're going to generate immune response to it, so you neutralize it later. So it can't be used for multiple injections or multiple vaccines. So it's a combination of the packaging and the envelope. And what we did is we found ways to make a synthetic envelope so that the advantage of the replicon could be targeted with a synthetic envelope. So what replicons are the genes of interest combined with non-structural genes that are rapidly assembled into a self-amplifying RNA and then injected into the body. And the advantage is they can be injected in very, very low amounts. So where a typical Moderna vaccine is 100 micrograms, we get potent T cells with as little as 1 microgram, and our standard dose is something around 5 micrograms. Again, immunotherapy for all, cost of goods, global health. You want to use a technology that is cost efficient as well as safe. And if you reduce the dose, you get both of those aspects. So I'm sorry, these are just for the people that are very interested in molecular biology, the aspects of alpha virus replicons. And alpha virus are the class of viruses that we chose to develop self-amplifying RNAs. What are the trade-offs of replicon delivery systems? Basically, if you're going to self-amplify the RNA, you have to be able to deliver it in a way that's safe. And the challenges have been with self-amplifying RNA is that when they're delivered with the standard formulations, which are called LNPs, which Moderna uses, et cetera, they're quite reactogenic. So these are studies from Arcturus, a study that a company that has developed self-amplifying RNAs. And they were the first ones to show that you could get a 20-fold dose sparing. Why? Because the RNA multiplies within the body. And so it's more efficient. So give a 5 microgram dose as opposed to here to a 100 microgram dose of Moderna. But there were issues with reactogenicity, and that's what I meant by the limitations of self-amplifying RNA. If you deliver it in a way that is not controlled in terms of migration within the body, then you get adverse events. And in fact, Arcturus hasn't been able to surpass 5 micrograms, in some cases 10 micrograms, without serious adverse events. So what are the challenges that we set out to address in our development of a better platform? One, use self-amplifying RNA because of costs and potency. Remember, T cells, lower amounts. But then to deliver it in a better formulation that did not have the reactogenicity issues. It doesn't do you a lot of good to develop a platform that causes severe side effects, or more importantly, can actually lead to RNA that migrates to tissues in the body where it shouldn't be. I'll talk about that in a few minutes. So we developed a platform we call Lion, and Lion with the rep RNA is very, very well tolerated in thousands of people so far. We've gone up to 25 micrograms in people, way above any dose that self-amplifying RNA platforms from other sources have gone. And in fact, in non-human primates, we've achieved doses of almost milligram quantities without side effects. So the RNA itself is not toxic. It's not reactogenic. It doesn't cause problems. It's only when it travels to tissues like the heart, where you get myocarditis, and you've read about this in the context of Moderna Pfizer vaccines. You want a formulation that stays in the muscle, stays in the skin, wherever you inject it, and it does not migrate to essential tissues to create a situation where your body thinks that the heart is now a foreign tissue because viral RNA is expressed there. All I'm showing here is that our side effects in India, Korea, Brazil, in the US where we've done clinical trials are all of the mild side effect natures. In fact, we have the only self-amplifying RNA platform that was ever approved for human use, and that's a COVID vaccine approved in India. These are just data talking about other self-amplifying RNAs. The Imperial College and my friend Robin Shattuck developed one as well. But the dose limitations of delivering these in LNPs stands for lipid nanoparticles, which are the particles used by Moderna, by Pfizer, by BioNTech, by CureVac, by Imperial College, by Arcturus, they all have issues with more severe side effects because they travel around the body. I won't talk about our data showing increased safety today in any great detail, but the paper will be out soon to show the mechanisms of why lion formulations do not travel. So the innate immune response is what we're trying to activate early. So you may recall that the immune responses are broadly categorized in innate and adaptive. I'm an adjuvant person, been working on adjuvants for 40 years, and we all focus on signals and formulation technologies that can activate innate pathways, whether they be through TLR or RIG-I. And these innate pathways provide the adjuvant effect that give your immune response the ability to respond rapidly. But they also are important, these adjuvants are also very important to give you an adaptive immune response. And the innate immune responses are wonderful with RNA because the RNA molecules themselves stimulate TLR3, TLR7, TLR8, in some cases RIG-I pathways, so they can be very potent, but again, they have to be controlled with the formulation. So why am I focusing on the safety aspects? Some of my colleagues say, well, if you're developing cancer vaccines, safety is not of primary importance, but that's absolutely wrong because, as I'll show you in a few slides later, we're working closely with the PREVENT Network at the National Cancer Institute in the US to identify patients early on so that they can be given prophylactic vaccination, if you will. The tumors are there, but they're precancerous lesions. Or in the case of a post-resection situation where you're trying to prevent recurrence, metastasis, et cetera, you really want a cancer therapy that can be administered multiple times as a maintenance therapy. And this is a novel concept for immunotherapy for vaccines because in infectious diseases, we rarely think about that. You might get a vaccine like, for example, shingles where you get it 2 times, or pediatric vaccines 2 or 3 times in your life. But for oncology, we have to have a very, very safe platform and a very, very potent platform. And those are hard things to combine. Usually with adjuvants, potency is associated with reactogenicity or toxicity. So what we're really trying to do is combine the 2 aspects of safety and potency together. And in this slide, we've shown that in these 2 slides, we have aspects of safety that are measured by type 1 interferon in humans and non-human primates. And serum interferon is a surrogate that we use for predicting safety of a vaccine. And we see that the doses of serum interferon go up with the amount of RNA, but we can get very potent T-cell activation response with very low doses of interferon as well. So this is the LION technology that we use. I developed this with the team about 8 years ago as an alternative to LNPs. LNPs are, as you see on the right, are nanoparticles about 100 nanometers that are produced with the RNA inside of them. So already you can see a complicated process for manufacturing. They have to be then frozen to be stored and shipped, and of course this leads to a high cost of dose for the vaccine. The LION particle was developed to be able to absorb RNA on the outside. And why is this important? It's important because then you can, I'm talking about practical aspects here, immunotherapy for all. You can produce the nanoparticle in massive amounts, store it for years at 4 degrees, and then mix it with RNA on demand. So it's not a co-formulation process from the beginning. Now think about this for oncology solutions. My vision is that each cancer center can have the formulation of LION ready to go when the RNA is produced then for a particular cancer vaccine. Then it can be just mixed in the clinic and injected into the individual. And that's how we've performed our COVID trials around the world. Mix and shoot. Very simple. So we think the future is to have maybe not personalized completely or maybe just semi-personalized, but RNAs that are chosen for that individual. That's why I'm so excited about to be in this particular session and the work that Dr. Davis talked about. We need to understand the genetics of a tumor so that we can design more optimal RNAs for different populations. And it's not that hard to do once you have the systems in place. And the flexibility of the Replicon platform together with the Lion technology gives us that flexibility. These are just some of the aspects of the Lion technology, and the different colors represent the fact that we've got a family of these things. They can be used for intravenous, for intradermal, intramuscular. They're different. It's all about the chemistry. For those of you who are interested and knowledgeable about surface chemistry more than I am, it's basically a cationic chemistry. You have to choose lipids and oils that are compatible with human use, that are available in large amounts, that are stable, and that can be safe by a variety of delivery routes, and that's what we've done. So as you see for the third bullet point here, Lion is the only delivery system clinically shown to be safe and effective in delivering RNA doses high enough to prevent and treat cancer. So what do we need in oncology? And the term cancer vaccines was popular in the 1990s. It became less popular when the vaccines continued to be disappointing. Why? Because the technology really wasn't there to make full-length proteins. These proteins are very difficult to make in E. coli and yeast in cell culture. So people were basically using peptides. And peptides aren't too immunogenic, especially because we didn't have good adjuvants to induce CD8 cells. So cancer vaccines are coming back, I will predict. You might call them therapeutic, you might call them prophylactic. I call them prophylactic because we're trying to prevent death and recurrence. Okay, they're preventative vaccines. Not so much in terms that we use today, like in influenza, we were trying to prevent it in the first place. But you have to have a safe and flexible platform. I've already talked about the biodistribution. This is well known. The biodistribution of LNPs is not amenable to repeated dosing that you need for oncology, but the Lion is. So we've done a lot of different types of studies in small animals, large animals, and in humans to reduce reactogenicity and enable these for safety for cancer. This is a terribly busy slide, but look at some of the immune correlates that we've identified with this Lion platform. If you look at the lower right, the interferon gamma production in terms of ELISpots is the highest with the Lion technology, and this correlated with protection against COVID in animals. What I'm getting at here is that the platform induces a broad B and T cell response profile that can be good for both antibodies and T cells, and I'll tell you why that's important in a minute. The other aspect of the Replicon technology is you can custom make your vaccines. You can combine several antigens. So Dr. Reyes just talked about, to me it was a learning experience, a different kind of melanoma. What if we need to have a different cocktail of melanoma antigens for one type of disease versus another? Again, a combination of genetic information, genomic information, sequence information from that patient, combined with then what the antigens you have ready to go. You can have a variety of melanoma antigens already made off the shelf, and then you combine them and mix them for that particular person. Can you do that with RNA technology today? No, you cannot, because with the typical COVID RNA platforms that are out there, you can exceed 100 microgram dose. And since melanoma antigens, for example, aren't that potent, you're going to have at least 20, 30 micrograms of each one. You want to combine several, you're going to have a platform that is very safe. And that's what we've shown here. Here we've combined 5 different viral vaccines and 5 different RNAs, and they're all ready to go. You mix and match as you need for cancer. In this case, we showed it, the context in RSV, COVID, and influenza, so you have a multi-respiratory disease vaccine. The point is, we've been able to immunize animals and get protection against all of these different vaccine targets. And this just is a proof of concept in humans, the COVID vaccine. In the middle, you see our T-cell responses that are quite high. And in the left-hand side, what was just published 2 weeks ago in the New England Journal, showing that almost no T-cell boosting with today's RNA vaccines. So the HTT platform on the right side gives you good antibodies, as well as on the left side, a good boosting of T-cell responses, in some cases up to 20-fold. So that's why we talk about building the world's most advanced cancer immunotherapy program. And just to finish up, I'll tell you what we're doing here. Again, the platform allows you to use high doses of multiple antigens, and we've started about 3 years ago a program with the National Cancer Institute in the U.S. to take all the cancer markers they've been working on over years and apply them to our platform technology. We started this, as I said, about 3 or 4 years ago. Well, 2018, more than I thought, 5 years ago. We have multiple academic collaborations. We'd like to expand those further. If you look on the website of the NCI under Prevent program, you'll see what it's all about. Early screening, early detection, in innovation, in innovative interventions. One of the targets, this is my last slide, is really talking about breast cancer. Many of you know the HER2/neu, for example, is a target for Herceptin antibody, and it's been used for years now. But what we didn't know is that many people on Herceptin can develop brain tumors in spite of being on the drug. Well, it appears that those are people that have low T-cell responses. T-cells can get through the blood-brain barrier, antibodies not so much. So with the NCI, we're developing a program to use a T-cell vaccine for HER2/neu to complement, not replace, complement Herceptin. And in South Africa, if you have low, we had a conference here, we said people that don't have access to Herceptin, we can actually induce Herceptin with a vaccine. So we can mix and match and design the vaccine according to any need. So thank you for your attention. I'm sorry to be over time.
Thank you to all our speakers. Maybe they can allow us maybe 5 minutes for questions. We over time. I don't know if there are any questions from the audience.
We have done not RNA-seq, but we have expression profile with NanoString platform that we are using. So for this data, we have also expression profile of.
There are lots of other tumors where there are identifiable mutations. For instance, non-small cell lung cancer, where the mutation would be an immune response to which would be more targetable. Are you also looking at those other areas?
Yeah. So thank you. I highlighted HER2 because it's a very well-known validated target, so it's a great proof of concept for the platform in general. The primary programs we have at HTT are breast cancer, prostate, and pancreatic right now. And the pancreatic is exactly what you're talking about, a combination of KRAS mutations with Johns Hopkins. So right now we're interested in just optimizing the platform and then applying, as you say, to other targets.
Yeah, thank you. Especially to the RNA vaccines. I've been working for quite a while on therapeutic vaccines in cervical cancer. And we also tried RNA and showed that by infecting cells with RNA, we got into the T-cell arm by MHC class 1 presentation, what we would expect. And we ran clinical trials and we didn't see any efficacy, despite great immune responses, T cells, cytokine expression, release, and so on. And then we looked at the MHC makeup of the tumor cells and we saw complete deletion of MHC class 1 on the cells. It's a major problem in immunotherapy of cancers that they have these immune evasion mechanisms. How would you ameliorate this problem? Because just inducing T cells is one thing, making them effective the other. And we haven't seen here the Clinical studies yet, of course, you didn't do them to the point that you show efficacy in the tumors in the patients.
So efficacy in immuno-oncology is largely dependent on the stage at which you get you apply the immunotherapy. But I look upon it as and the recent data that we're getting out of pancreatic tumors, even with peptides, are very promising in terms of very good immune responses. So it's a matter of staging, but it's also, I look upon it with as a complement to checkpoints. I mean, if you can reduce the immune suppression with a checkpoint inhibitor and get 20% increase in efficacy, you should be able to amplify that effect further by expanding relevant T cell populations.
Yeah. Thank you. Again, this is the T cell arm. It's the effector arm, but not the receptive situation of the tumor cell, because a tumor cell that has no MHC faced with the cytotoxic T cells will not be killed. That's the problem. And if you start out with early tumors, you might catch, before you have this deletion process of MHC, the selection process, but you also might enhance the selection process. I just want to point this out because it's very important to not forget this. We know it, but we should also put it into consideration with the studies and with the design of immunotherapy. I have been very frustrated with immunotherapy. I've given up when the preventive HPV vaccines came on the market, and I did my immunology there and contribution, but checkpoint inhibitors, for example, like in 10% of cervical cancers, they have an effect. These are those where the MHC complex is methylated and switched off and can be re-expressed. But in those cancers that have a deletion, I'm very pessimistic. Still, 10% is a good number in cancer therapy, I think.
We over time, maybe just like one last question and then unfortunately.
Question to you.
There's two more.
A question to you, Dr. Melissa. Thank you so much for the very interesting and obviously painstaking work that you guys have been doing with disparities as far as the African American and how you could show the difference between American Africans and then folks who are based here in various other countries. My question really was talking about triple negative breast cancer, and you could show the differences. Do you have any solutions for that? It may be an easy question, but may have a difficult answer. So please, I'll be perfectly comfortable if you say, well, looking at various issues, or you already found something.
Yeah, so we have a couple of potential drivers of mutational processes that we're modeling right now. One was the target of a GWAS study that actually the European allele is a protective variant, and so it has been under research. So we're looking into that. Also, because of the immunological landscape, we're thinking something around immunotherapy checkpoints. So how can we identify the triple negative breast cancer patients who would be more, beneficial for immunotherapies? One of the things that we note is that the clinical trials for immunotherapies, where there hasn't been a great success for immunotherapy in triple negative breast cancer, as other types of cancers, have been devoid of people of African descent. And so if we sort of expand the catchment of those clinical trials, and we may actually see that this population of individuals could benefit more than others. The Duffy-Null, mutation as well, chasing that down in terms of the systemic inflammation and how it impacts the activity of the immune cells. But then lastly, the thing that's in the cancer discovery paper that I didn't necessarily talk about today are the subtypes of triple negative breast cancer. So redefining what a triple negative breast cancer tumor is in the context of those ancestral informative markers. Thank you.
But just for curiosity, that is also a blood type on RBC, as you have probably mentioned. Is it the same DUL that we're talking about or something different?
More time.
The
Dupinol.
Yeah, that is also a blood type.
Yes,
if I'm not
absolutely.
mistaken. Is it the same, right?
Yeah, it's
Just went back to medical school. Thank you.
If I can continue in that topic, I had a question. So I remember, like if it was today in my genetic classes, that my professor was mentioned that in a small African village, you have more genetic diversity than in Europe. In that context, the migration that went to the American continent doesn't represent the whole diversity of African population. So my question is, do you think that studying only Black American, you can miss a lot of diversity that could be important for the treatment of Africans from different regions?
So that's a great question. And I think, so when we think about the diversity in Africa being greater than other populations, we're talking about a broad human, landscape. We're talking about across the human spectrum. Specifically now what we're talking about are a subset of people who have a shared disease. And so in that way, we've sort of distilled down that diversity that we're talking about that kind of makes it too complicated, if you will, to identify the genetic mutation in the spirit of GWAS studies, right? So we're talking specifically about a group of people who have a shared disease and a shared genetic background. Yes, there's a lot of complexity in that background, but they clearly across this population, across the planet, are sharing something similar to the Ashkenazi Jewish mutations in BRCA. There are EGFR mutations in Asian populations. We see that same level of diversity, of course, when we aggregate Asian Pacific Islanders, but when we start to disaggregate based on their genetic background, we can see an enrichment for those particular mutations. So that's sort of the concept. I don't have the mathematical model yet. I need to talk to a mathematician, but I think that, I agree. Like there's a lot of diversity, as I just showed, in African Americans, which makes it all the more difficult, which is why it's all the more important to define what that diversity is. But thank you for that.
My name is Lungi Mkiza Kwichana. I'll be chairing this session, and the theme is Precision and Translational Oncology. And what an opportunity after the previous sessions where we've seen the diversity, and actually we'll see how the utility of the Human Genome Project, harnessing the power of artificial intelligence, can actually bring us to the clinic, actually, because the ultimate is the clinic. So we have speakers. Our first speaker is Professor Olivia Elementos, armed with the qualifications in mechanical engineering, 2 masters actually in mechanical engineering and in artificial intelligence, and then a PhD in computational biology. He has quite an illustrious career, and I will just highlight a few. He is the director of the Englander Institute for Precision Medicine, and he also is an associate director of the Institute for Computational Biomedicine. He has achieved numerous honors, including the co-instructor and co-organizer, Cold Spring Cancer, Walter B. Winston Award, and the NSF Career Award of the the NSF Career Award and Genome Graduate Fellowship for Outstanding Students. He has multiple roles in this sphere, and in the interest of time, I have just summarized his illustrious career, and he is going to give us a keynote talk on the future of precision oncology. Over to you, Professor.
Thank you so much. All right, it's a real pleasure to be invited to speak with you guys today. I wish I could be there in person. I've been to South Africa before. It's an amazing, beautiful country. I would have loved to come back and hope to come back in the future. So the title of my talk today is going to be about the future of precision oncology. And what I want to give you a sense of is some of the work that we're doing at Weill Cornell Medicine and our Institute for Precision Medicine in the area of using technologies to assess cancer samples and using information from the results of this technology to personalize cancer medicine as much as we can. These are my disclosures. This is the content of the talk today. I'm going to give you an overview of precision medicine as we do it at Weill Cornell. I want to talk about DNA sequencing, which is the workhorse of genomic analysis of tumors, but is also undergoing an evolution towards platforms that look at a very small number of genes, towards what we think is the future of the field and the ability to analyze the DNA of tumors from beginning to end and looking at every gene and every mutation. But I also want to highlight the fact that there's tremendous amount of information outside of the DNA, and we want to be able to capture that information. And so this is something I'll discuss and spend quite a bit of time, in fact, on looking at the ability to assess tumors at single cell resolution and learn a tremendous amount about which tumors are likely to respond to therapy based on this non-DNA assessment of tumors. So our institute is really looking to make medicine as personalized as possible. We believe that medicine has always been personalized, but there's amazing potential to increase the personalization of medicine, and that we think can be done using technologies. And I'll get back to this. Just to make my case, obviously this is an audience that I'm sure believes in the concept of personalized medicine. But just to make the case, this is an example here of 2 patients with breast cancer. And what you see in this slide here is the list of mutations in the DNA of the tumor cells of those patients, of those 2 patients, and some of the genes that those mutations are in. And what you can see here in this slide is that these 2 patients who have the same type of disease as we sort of diagnose and label the disease, in fact, have tremendously different mutational content. They basically have no mutations in common whatsoever. So we think that's really important, and we really are trying to use that information as a way to sort of guide our approach to looking at cancer and assessing cancer therapy response. We do this using technologies such as the ones that you see here on the slide, technologies to sequence genomes really quickly, technologies to image tumors at single cell resolution, and a lot of AI that allows us to integrate data from all these technologies. This is a highlight here of the precision medicine oncology workflow at Cornell, which starts with biopsies of patients, profiling of tumor biopsies or surgical samples, integration of data into reports, and highly personalized recommendations. And I want to highlight that we do this in the context of research and enroll patients into research protocol to be able to do as much research as possible on the gigantic amount of data that comes out of these analysis. The field of analysis, so DNA analysis of tumors, as I say, has been undergoing a dramatic change, moving from targeted panel sequencing, looking at a small number of genes, like a very tiny window on the tumors, towards whole exome sequencing, which is looking at every single gene. And I think what we need to do moving forward is whole genome sequencing, essentially ability to sequence genomes from beginning to end, looking at every possible mutation. So this is a journey that we've taken at Cornell. We actually have implemented whole exome sequencing as a clinical tool. We learned a tremendous amount in the past about cancer genomes. We learned, for example, that they're very complex. Many cancer genomes have a large number of mutations in lots of different genes. And the challenge has been to essentially identify among those many mutations which ones are most important and potentially have the potential to guide therapy. I'm showing here an example of a prostate cancer patient with an advanced sort of stage of a disease. And this is a copy number alteration plot looking at different sort of gains and losses of mutations across the genome. And as you can see, there's a gigantic number of such mutations. There's a tremendous challenge in finding the important mutations in those. So we've actually taken on this challenge in the past few years, been able to essentially automate the production of reports, individualized reports, such as the one that you see here, and essentially go back to every patient and contain essentially all of the different mutations that we find in each of the tumors that we sequence. But as we have been sort of doing this for some time, we realized that we need to go beyond just looking at the genes. We really have been embracing this idea of whole genome sequencing, which is sequencing tumors from beginning to end. We're part of multiple working groups that have been really looking at this approach in a lot of detail. How do we implement whole genome sequencing in the clinical setting? And we published this paper, which I highly recommend if you want to get a sense of the many advantages that come with whole genome sequencing as a clinical tool. This paper contains a table which I like very much, which essentially is looking at all of the advantages, all of the signals, all of the information that you can get from whole genome sequencing. And this table here in the last row here has essentially an assessment of all of the different pieces of information about the tumors that you can get from whole genome sequencing. Some of the ones that are really exciting to us are, for example, the ability to assess homologous recombination defect, which is essentially assessment of the ability of patients to respond to PARP inhibitors, which is something that's very important in the field now. We just realize that many patients have the potential to respond to PARP inhibitors. They may not have a BRCA mutation in the traditional sense, but they have something in their genome that confers essentially a BRCA-ness sort of phenotype. And whole genome sequencing is the gold standard for assessing such information. This paper here came out just a couple of years ago now. This was actually an assessment of structural variations in lung cancer genomes, looking at lung cancer tumors that did not have traditional driver mutations. Long story short, using whole genome sequencing, we were able to find a very large number of mutations missed by whole exome sequencing in targeted panels, many of which were actually structural variants, the kinds that you see here at the bottom on the right side, which in fact actually were impacting known lung cancer genes, but in a way that is not detectable using whole exome sequencing and whole genome sequencing enabled detection. And so this led to this work around whole genome sequencing led to an effort here at our hospital to deploy whole genome sequencing as a clinical tool for as many patients as possible. And this is a bit too early to sort of relay the results of this initiative, but this will be presented at AACR, one of the largest cancer conferences, and by some of the investigators on this slide. In the last, sort of remaining part of the talk, I want to highlight what I said. I think is really critical is to go beyond DNA to understand the makeup of tumors. Why is that important? Because we know that tumors are actually not only tumor cells. A tumor is a complex ecosystem with lots of different cell types. We believe more and more, and I think this has been shown now by many groups, that the composition of this ecosystem is really critical to the life cycle of a tumor, from initiation to progression, but also to response to therapy. And so we've again embraced this idea of being able to assess this microenvironment with as much detail as possible. How do we do this? There are lots of different ways by which you can do this. Basically, we really have decided to assess the spatial kind of architecture of tumors using technologies such as the one that you see in the middle here, which is basically a way to analyze tumors using single-cell proteomics. There's single-cell transcriptomics, there's other types of technologies, but the one that we use is the one in the middle, which is shown here, is ability to assess tumors using single-cell resolution proteomics, using antibodies, targeting specific markers, for example, is expressed only in certain types of immune cells or certain types of stromal cells, and assessing essentially the amount of the antibodies at different locations within tissue sections. This is a technology called imaging mass cytometry, which Dr. Davis has also discussed, and is very powerful, and we've been using now for many, many different types of cancers. This is what the technology looks like here. We have 2 of these machines here at Weill Cornell. We use them on different types of tissue. A lot of what we use is tissue microarrays, which is what you see on the right side, with the ability to assess lots and lots of patients on the same slide. Each spot on the slide here is one different patient. And so, as you can see, there's multiple patients on this particular slide. We've applied this to multiple different tumor types, including a tumor type that we've been working on for some time at Cornell, which is a type of B-cell lymphoma, which is very aggressive. It's called diffuse large B-cell lymphoma, treated uniformly with the same type of chemotherapy, but we know that there are responders, non-responders of this kind of therapy. And it's been a challenge in the field to identify ahead of time at diagnosis responders to therapy, with an initial focus very much on the tumor cells. This analysis that I'm going to show you here was actually try to focus on the microenvironment of the tumors. We looked at 400 different patients in a cohort of patients that had a tremendous amount of annotation, clinical annotation, additional type of information such as mutations. And we learned a lot by applying this kind of single cell proteomics technique to this cohort. I'm going to try to give you a sense of what we learned. We assessed multiple markers. This technology here is using markers that you have to choose ahead of time. You have to design antibodies to be able to identify where specific markers are expressed in the tissue sections. These are the markers that we use in this analysis. Immune markers that tell you where you have immune cells on the tissue, functional markers that tell you what kind of cells you have, including what kind of tumor cells you have, and structural markers that tell you something about blood vessels or fibroblasts and so on. The data that comes out of this kind of technology is really amazing. This is an example here on normal tissue, normal lymph nodes, where you can really see individual cells at single cell resolution and assess using markers, for example, the proliferation, whether the cells are proliferating or whether they express specific checkpoints. Pretty amazing ability to essentially understand the battlefield within tissues and within cancer tissues, and understand how tissues contain cells that interact with each other and potentially important for responsive therapy. This is what the data looks like, looking at individual cores, the cores that I showed you earlier in B-cell lymphomas. You can see some structures, you can see blood vessels, for example, here with CD31 expression, as well as other types of markers in those samples. The analysis of this kind of data becomes a computer vision analysis where we have to find the cells. We have to quantify the signal within each of the cells. Once we've done this, and this analysis, in this particular cohort gave us about 7 million cells in total, so it's a lot of different cells. We can assess, for example, the identity of each of the cells, whether a cell is a B cell, a T cell, a tumor cell, and that's what we did here. And so we are able to assess the overall global identity. But we can go down to subpopulations of cells, for example, subpopulations of B cells. Again, this is a B cell malignancy, so we see a lot of B cells in this cohort, as well as subpopulations of myeloid cells with different markers that you see here. Different abundances across different tumors, subpopulations of stromal cells, subpopulations of T cells, and so on. As you can see, this is a very rich sort of picture here that you see. A lot of it, again, is focused on the microenvironment. So now each of the cell that you saw, thank you. Each of the cell that you see earlier has an identity, and what you can see on the right side here is essentially each dot is one cell with a color that corresponds to the identity. And so, for example, the green cells are the B cells, and the red cells are some of the T cells. So what we can do with this kind of analysis now is actually focus on trying to find subtypes of microenvironment. This analysis was very focused on microenvironment. What we did was to actually try to assess whether there are actually multiple different subtypes of microenvironment in this kind of disease here. And to our surprise, we were able to find 5 different subtypes of microenvironment. One of them was a cold microenvironment with very few immune cells. We had essentially a gradation. We had a CD4-rich microenvironment, an inflammatory microenvironment, and a cytotoxic microenvironment with a lot of cells expressing coenzyme B, as you can see here, and that's what we call it cytotoxic. So, this was essentially an observational picture of a microenvironment of this disease, which had been very poorly characterized in the past. This is just to show you what the microenvironment looks like in the actual samples, and it really corroborates the classification. I think what's really amazing about what we saw here is that, and we didn't expect to see that, is that there was a very strong correlation between the type of microenvironment that these tumors had and response to chemotherapy in those patients. We saw actually two subtypes of microenvironments, the cold subtype that I showed you earlier, as well as the cytotoxic subtypes, which had very poor response to chemotherapy. Patients with this subtype of microenvironments, irrespective of mutations that they may have, actually do very poorly when it comes to response to chemotherapy. So what we did was to take essentially all the signals, this information to the next level, and build a machine learning method that is able to predict survival to chemotherapy for those patients with actually a tremendously high level of accuracy, much better than anything that has been used in the field so far. It's just an example of how the combination of this kind of single cell imaging, as well as some clinical factors, as you see here, like age, was really able to classify tumors into non-responders and responders in a way that we didn't expect to happen. And so we're trying to go beyond just this kind of essentially assessment of the abundance of different cells within tumors, looking at compartments, looking at the architecture of these tumors, looking at whether there are compartments in those tumors that could potentially reveal something about the biology of these tumors. Dr. Randero in my group, who now is leading his own group in Vienna, has been putting together a very nice computational method to essentially find those compartments I was telling you about. I don't have time to go into details, but the idea is to integrate data from proximity between cells and the cell types of each of the cells within one of the images that I just showed you. We demonstrated that we can use this technology this technique to recover compartments in normal tissue, for example, the lung tissue with tremendously high level of accuracy. We applied this to tumors, including bladder tumors, for which we had a manual annotation of the different compartments, and likewise so that this technique is very good at finding those compartments. In breast cancer, where we had also a very large cohort, we're able to show that this compartment structure revealed that this technique that I showed you about, essentially finding different compartments, such as, for example, areas of the tumors that have a lot of stromal cells and a lot of T cells. This kind of compartmentalization of the tumors in the microenvironment reveals something about survival upon chemotherapy or treatment, such as surgery and radiation. And we find different types of compartments in those tumors that have correlation with response to therapy. So again, this is data that shows us that there's a lot of information within the microenvironment in terms of compartments, in terms of abundance of immune cells and other non-tumor cells, that is really critical that we need to capture to be able to provide the best way or the best level of precision medicine moving forward. And so I'm going to stop here. This is the end of the talk, and hope that we can continue the discussion of our questions. So thank you for your attention.
Well, thank you. We have then Prof. Lolas. I think because of the time delay that we've had, maybe Prof. Hicks had other commitments. Prof. Lolas is going to be our next speaker. He is the coordinator of the Systems Medicine Group in the Oncology Unit of the 3rd Department of Internal Medicine and Laboratory of the National Kapodistrian University of Athens. He is also the founder of the startup Incelia, which focuses on developing the multi-scale mathematical models for the designing of in silico therapies in a personalized and precise manner. His group has already developed mathematical models describing complex processes such as tumor-induced lymphoangiogenesis and proteolysis. Lately, they have moved their attention to developing in silico models that are related to the emerging field of cancer neuroscience. He's also going to give us a talk on cancer neuroscience titled
Thank you very much. I would like to thank the organizer.
Deciphering the Roadmap of Cancer Neuroscience.
Thank you. Thank you for the introduction. And thank you also the organizers for giving me the opportunity to give a talk about cancer neuroscience. As you mentioned, I'm a mathematician, so I'm coming from a different background from the rest of the speakers. So I will give you a small presentation regarding the mathematical models that we have done in the past and what we are aiming to do in the future. So an outline of my talk, it has to do with the phases of cancer. I will talk briefly about the blood and the lymphatic vessels, and then I will focus on the neuronal system and the future actually directions that we have in the oncology unit regarding translational oncology and personalized medicine. So most of you know that the cancer becomes lethal when, for example, it invades the extracellular matrix and can metastasize either through the blood or the lymphatic system. It was 50 years ago that actually Judah Folman, he did the first experiments in tumor-induced angiogenesis when he put tumor cells in the back of the eye of a rabbit, and then he saw that tumor secretes specific angiogenic factors, and the blood vessels, they start forming a network approaching the tumor. So it took a few years, actually, the mathematical modeling community to develop models related to angiogenesis. And it was back in 1998 in the University of Dundee when Professor Chaplin and Sandy Anderson, they did this model where they put the tumor cells at the bottom of the cube and the blood vessels on the top. And you will see that they can manage to capture anastomosis, loops, networks of blood vessels and angiogenesis. For several years, although a lot of the community, they focus on the blood vessels, the lymphatics didn't have so much attention. And this was because actually they couldn't find specific markers for the lymphatic vessels. They thought that the lymphatic vessels were just passive participants of the tumor growth and tumor dissemination. It was 1999 that your own actually Michael Pepper and other groups like Michael Dertman, Michaela Skobe, and Jiha Ikao and Karolinska Institute, they investigated the effect of VEGF-C and VEGF-D in VEGFR3, which is the receptor in the lymphatics. So a few years later, myself and Professor Friedman in the MBI Mathematical Bioscience Institute in Ohio State University, and Michael Pepper, we developed the first mathematical model that has to do with tumor-induced lymphangiogenesis. We also proposed, ahead actually from the experimentalists, that they should also focus on and pay attention on the role of the urokinase plasminogen activation system, and especially plasmin, since plasmin can cut VEGF-C and make a mature form of VEGF-C that can bind with high affinity to the receptors of the VEGFR3 in the lymphatic surface. So, as I wanted to show you that takes some time for the experimentalists and also the research community to address and also to accept that there are other parts of the body that play a crucial role in tumor growth and tumor dissemination. This is actually the case with the neurons, although everybody knows that perineural invasion is very important and neurons are around, the tumors. Nobody has looked, at least recently, on the effect of neurons in tumor growth and tumor dissemination. But before I continue, I just want to mention a little bit about the wording. Everybody knows about neuro-oncology. That is actually a term that it deals with the brain tumors. Neuroimmunology, it has to do with the inflammation of the brain. But cancer neuroscience or exoneural biology, which I introduced in the first slide, it has to do with the phenomena that there are outside of the nervous system, of the confines of the nervous system. So it was back in 2006 that Frank Henslade, he was the one that he introduced the term neoneurogenesis, meaning in the same aspect like angiogenesis and lymphangiogenesis, that tumors secrete specific factors that they make the neurons either to elongate or to produce new axons or new neurons. It took a few years, for the experimental community to do the first experiments related to tumor-induced neoneurogenesis. It was in Bordeaux, where Claire Magnon, she did the first experiment in vivo related to the effect of the neuronal system, actually the autonomic nervous system, to prostate cancer. She was able actually to capture that there is a complementary effect between the sympathetic and the parasympathetic system in tumor growth and tumor dissemination. In 2019, it was the first gathering of the Neuron Tumor Society that they actually try to identify the roadmaps of cancer neuroscience. So they had four pillars, and they mentioned that somebody that will be interested, for example, in investigating the neuronal aspects of cancer growth and cancer dissemination, they should focus either in the electrochemical signals between neurons and cancer, either in the autocrine and the paracrine effects of neurons and cancer, or how, for example, the neurons, they affect the immune system, or how the treatment affects the neuronal system. So it was back in 2015, 2016 that myself, Arianna Bianchi, and Professor Sirigos, we developed the first mathematical model of tumor-induced neoneurogenesis. So we built a model based on what Claire Magnon has done in her first experiment in 2013, having in mind both the nerves from the sympathetic and the parasympathetic system, plus cancer cells, and several factors like nerve growth factor and axon guidance molecules. I'm not going to go through with the mathematical models, but I just want to tell you that we went one step further and we were able to show that chronic stress plays a crucial effect on tumor growth. I mean, even if we start with lower, let's say, cancer cells or lower, tumor volume, if we have a lot of chronic stress, a lot of norepinephrine, this gives the opportunity to the tumor cells to grow and actually disseminate. Since we talked about the chronic stress, it was after a few years, actually in 2016, that Erika Sloan from the Monash University, she presented the first experiments that had to do with the chronic stress and the effect of them in the lymphatic system. So she managed, for example, to capture the cancer cells in a cage for several hours, and then she was able to see that the cancer cells, for example, actually, due to the stress effect, they secrete norepinephrine. Norepinephrine can activate prostaglandin and COX-2, and then this can activate VEGF-C, which VEGF-C relates to the dilution of lymphatic vessels. So it's much more easier for the cancer cells, for example, to enter the lymphatic vessels and then metastasize. We are actually developing currently a mathematical model that's trying to build and take together actually the macrophages that Erika Sloan proposed, the tumor cells and the lymphatics, plus also investigating the role of VEGF-C, prostaglandin, and norepinephrine. In the model that we have produced, we were able to capture the competition for space that nobody has looked, for example, before in the experimental evidence. I mean, tumor cells, lymphatics, plus macrophages, they compete with each other, who's gonna take the space that they have around them and how fast they can do that. So, and by making this hypothesis, we were able to see that, for example, when we have the effect of stress, we have an earlier onset of tumor cells. Also, macrophages with stress, they start beginning to grow even faster. And the same goes for the example with the lymphatic system. I'm not going to show you more of the results. I just want to tell you that there are a lot of open questions between the effect of neuron system in cancer biology. Although the perineural invasion, as I mentioned, is a very well-known phenomenon, there are a lot of molecular aspects that nobody has highlighted until now. Also, there is a big gap in the experiments, between how the neurons, for example, they can affect the immune system, macrophages or fibroblasts, and also if we target, for example, the neuronal system, what will be the effect on tumor growth? So what we aim to do in the oncology unit and what we have started is trying to analyze tumor biopsies and see the nerve density in both in the non-small cell lung cancer and small cell lung cancer, and see if there is actually correlation on the tumor outcome and the aggression of the tumor. So apart from that, we have started doing PDX models, taking tumor samples from patients and sending them for doing zebrafish experiments. Zebrafish is actually an animal which is high throughput, so we can estimate, so we can see how the tumor cells, for example, they travel, they metastasize, and we can estimate how many of them they have already metastasized from the primary tumors. Also, we have started using the zebrafish experiments, and we have started, testing several drugs, either in combination or alone, to see what will be the effect on the specific tumor sample from the specific patient. So last but not least, what we wanted to do is bridge the mechanistic models that I have presented, based on the experimental evidence of zebrafish experiments, together with tumor biopsies that we'll analyze using AI algorithms in a new, let's say, setting, using a Bayesian approach from the mathematical point of view, and give a decision, let's say, support to the clinicians. So I will close my talk now, and I would like to thank, these are my collaborators around the world, that they have helped me, producing this kind of research. And thank you very much. I will close with this cartoon that has to do with the predictability of the mathematics and of science. Thank you very much.
Thank you very much, Prof. Lalos. Thank you very much. That was very illuminating. Back at school, we used to, when we ran out of answers for etiology of any diseases, we would put stress, and we didn't know how far it went. Thank you very much for that talk. And again, we will ask, we apologize again for running this late. We will ask our attendees to please pose the questions. I'm absolutely sure there are thousands of questions that would have come up, but in the interest of time, we would like to release you, and thank you very much again for this illuminating talk. We highly appreciate. Thank you. Okay, we will start now with Professor Godfrey Grech. He is an associate professor at the University of Malta, focusing on oncology research, including breast, colorectal, lung, and cervical cancer. He was awarded a 3-year fund within the Department of Pathology to study biomarkers that are predictive of early metastatic disease in colorectal cancer. He's highly recognized by the clinical sector and runs numerous projects in collaboration with Mater Dei Hospital and international institutions. Of note, his research led to novel predictive biomarkers in triple-negative breast cancer and the optimization of a multiplex RNA-based expression assay to molecular classify breast cancer patients. Over to you, Prof. Grech. I have not completed your bio. It's in here in the interest of time. Thank you very much for joining.
Okay, so thank you very much for coming on in time after lunch. So today my talk will focus specifically on copy number variations that we actually read via RNA expression. So when I say RNA expression, in this case would actually provide us information about the presence of CMVs. And we use this as an endpoint measure for in blood-derived exosomes, and I will explain what we exactly do for that in both breast and colorectal. So I'll take you through a history of how we analyze these CMVs in exosomes. There's also a joint project with industrial partner OmniGene Medical Technology, and is run by the University of Malta. So we run a number of projects. For instance, the IMPRINT and the IMPACT project. So the IMPRINT project looks at cell surface markers to try and identify these epithelial and mesenchymal or joint epithelial mesenchymal cells. Also because when we try and isolate circulating tumor cells from blood, we know that we don't capture all of them because they will not express high levels of EpCAM, which is the main antibody used to capture circulating tumor cells. So we embarked on this project to identify cell surface marker in various cell line models and also in patients. And thankfully, 2 weeks ago, and I'm going to announce it here, for instance, we actually found one surface marker which is present on a cell model, which previously we couldn't pull down with spiking experiments. This is the triple-negative breast cancer cell line 231 for those that would know that. So then we have also the IMPACT. The IMPACT is more basic research trying to find biomarkers, novel biomarkers to put together to run into our high throughput multiplex assays. And in fact, as part of this project, which started a long time ago actually, we also identified a new subtype of triple-negative breast cancer patients, and we have already filed a patent on the list of these biomarkers. So although most of our work is cell line models and also in terms of basic research, and also having applied and validate our biomarkers on cohorts, patient cohorts using FFPE, formally fixed paraffin embedded materials from the pathology department, which are also part of our research teams. 5 years ago, we started on the embarked onto the liquid biopsies, because now we have prospective cohorts in which we can take the FFPE material of consenting patients, and also we have a matched blood sample, and that matched blood sample is 2 × 9 mils. So definitely we need to increase the amount of blood, and that's why prospective sampling is extremely important. What's also very important is, that of course solid tumors are represented in the bloodstream by various methods, various characteristics, including circulating tumor cells, as I said before, but also cell-free DNA, cell-free RNA, and now also we're working on exosomes, and this is what we'll focus today. So if you have a blood sample, we have various materials that we can get out of a blood sample. From the plasma, we get cell-free, tumor-derived, including tumor-derived DNA. We take exosomes from plasma samples. We look at actionable mutations. We look at DNA methylation markers. But also, we isolate exosomes immediately from these cells so that we biobank it. And these exosomes are actually endosomal in origin. And when cells are stressed or under hyperproliferative states, they will release these exosomes. Now, it's important that in plasma, we also have normal physiological exosomes. So we also immunoprecipitated tumor-derived ones. So maybe you know about the ongoing sort of our FDA-approved circulating tumor cell methods and technologies. They mainly are utilize EpCAM antibody to capture CTCs, and we are now finding other types of cell surface marker so that we actually multiplex the immunoprecipitation using different antibodies as well for circulating tumor cells. Now, of course, every technology that we drive, because our research is in general basic research to find novel biomarkers, but also research on technologies to get them in line with the sensitivity that we require them. So, using cellular models, so we generated various tissue blocks and tissue microarrays from cell lines that they are very useful when it comes to actually validating and utilize it for quality control purposes. We can do sort of antibody staining. And for instance, here it is an EpCAM antibody. And you can see that most of the some of these cell lines do not actually express EpCAM. But looking at copy number variation, we looked into breast cancer, the HER2 sort of receptor. All right, and you can see here, for instance, a cell model which is very well known that overexpresses HER2 and is also SK19. So we can do all this kind of analysis using tissue microarrays on cell models as well. For instance, this particular cell line is totally negative of EpCAM, and anything similar to this cell line will not be identified in the bloodstream using EpCAM antibody. Now, of course, basic research taught us that here we have a specific biomarker in this particular cell type, and it is fibronectin. And from there, so we do a lot of gene expression analysis and RNA sequences. And here we found that one particular protein is extremely expressed in the cell model. And then we looked at our patient material, and in fact, we found that FN1, which is a normal stromal marker, from mesenchymal origin, then we find that tumor cells that actually express the FN1 cytoplasmically, all right, are associated with lymph node metastasis. So all this will put into our patient material and derive information that we require. So yeah, we do multiplexing. I will show you the technology in a few minutes. But basically what we do is we lyse the cells in this case, or FFPE material, and then we actually can measure more than 40. Actually, we have one assay which is 40-plex. We can read 40 gene expression at one go in the same lysate. Here it's a 6-plex assay, so there are differences. You can make different combinations. This is a 6-plex assay, which was used for the circulating tumor cells, including EpCAM and HER2, and others like estrogen receptor, cytokine 19, and FN1, because now we introduced that because of our knowledge. Now we'll use Luminex technology to read out these multiplex, because it is a bead-based assay which captures the target and amplifies the signal. And then it's read with these kind of instruments, which is the XMAP technology. Now, very quickly, the XMAP technology, which is a Luminex bead-based multiplex assays that can read up to 80 genes. I will tell you that now we have technologies that we are reading more than 200 antibodies in one cell lysate, so we are increasing that sensitivity in our labs. So the way it works, that you have the target target RNA, which has different colors here, okay? Then you have the beads that have specific probes on them, which bind to specific capture extenders, which actually capture the target RNA. Then there is the buildup of a specific scaffold on it through label extenders. And with these pre-amplifier and amplifier, then we can increase the signal. So basically, we are not doing target amplification here, but we are doing signal amplification, which makes a big difference in the input. With these kind of assays, we read low input, low quality material, even in terms of RNA. And multiplex, so we can read a number of targets as one go. So within these targets, when we have like a 40-plex, we divide them into those, we call them classifiers, in which we try and identify the cells that we have at hand. And those classifiers will give us information, for instance, on epithelial or mesenchymal origin. So these two markers, CDH1, which is E-cadherin, and EpCAM are epithelial. And these are the different breast cancer cell lines. You can appreciate here that this particular cell line is not expressing the epithelial markers, and also the 231 does not express these epithelial markers. Now, these were originating from primary cell lines, and these from metastatic cell lines. So because of scale, I've built it up, because otherwise we don't see the results. So in fact, when we start putting the mesenchymal markers, this particular cell line shows us that we actually have a cell surface marker that we can use here to capture them, and that's N-cadherin, okay? So it's a CDH2. And we also, as we know, there is the expression of FN1, in which we have huge studies ongoing right now to understand its association with invasion and metastasis. But again, 231, we don't have any surface marker which we can use to capture them. What we know that they are Vimentin positive, okay? And this, therefore, in one test, we are seeing all this information on this particular model at one time, in one run. But in addition to the classifiers, we also would see tumor sort of characteristics in terms of gene expression. Now, since the talk is about the CMVs and the cell surface marker to capture, here we have I included the ERBB2 information. So here, in fact, this particular technology is a Thermo Fisher technology called Quantigene. And what the blue lines represent EpCAM expression, the orange cytokeratin 19, the yellow FN1, and the green is HER2. And what I'm showing here is on this particular axis, this is the median fluorescence intensity that we get from the instrument. And this one is the input of cells in the blood. So we actually spike cells down to 15 cells per mil. And this was actually published in Methods in 2018. And as you can see here, we can actually capture the EpCAM on these two markers, but not as low as we would like. But anyway, we can have the HER2, which provides us with the right amount for capturing. So this is to synthesize the expression, how to capture the expression. So some answers and results. So we run these assays for diagnostic metastasis, EMT, and therapeutic resistant in colorectal cancer, and we've seen that amplifications, as we were expected in these cell lines, there is MET and EGFR in SW48 and MYC in COLO320. Then if we take these 18 plex and look at these particular diagnostic markers, we can actually have 3 genes that will give us the amplification and capture as present in mostly all of the patient material. So these are all patient material here, okay? And if we have TPX2, which is highly represented as an amplification in colorectal cancer, FZ21 and IGF2 are extremely important in primary results, primary tumors. But we know that TOP2A, 50% of CRC and lung metastasis, because we looked at primary tumor, invasion fronts, lymph nodes, and also metastatic, everything matched within a patient, and also metastatic lesions. We can now actually state that 50% in lung metastasis have a high TOP2A amplification. And cyclin D3 is amplified in 50% of lung cancer metastasis, so lung metastasis. And we have other overexpressions that are associated with specific metastasis. So very quickly, because I only have 1 minute left, okay, these are the exosomes. They are very small vesicles in the range of 50 to 100 nanometers. We capture them via various ways. This is a spike-in experiment using 2 particular commercially available kits. Although we have our own methodology, we also utilize other kits, put off 500 microliters only. And the results of the quantigene is basically we have a bead. We do a biotonated antibody to capture whatever we need. In this case, it's exosomes, because CD9. And then we actually capture the exosomes in an exosome bead complex. And we go with flow cytometry. And with flow cytometry, then we can use any antibody to see if the exosomes are there. This is the exosomes here, single beads, double beads. We also see exosomes coming together, the beads coming together. And that is the shift. So we can see here a shift in CD63 positivity in this particular cell line model, but not in this. And also using quantigene, we have actually utilized various commercial kits, and we find that this one was the best commercial kit in terms of spiking experiment, although today we move on to input more mils, because in this case you can only input 500 microliters. And if you look at the cell line and the exosomes, you see that there is a same expression, although it's lower in exosomes because it was only 500 microliters, but we can see the same trend, okay? So in this cell line, we have 19 and RB2 and here the same, whilst in the 56780, we see the FN1 in both cell lines and also exosomes. Now I'm going to conclude. So if we put the RNA on this axis and the exosome MF5s on this axis, we can see that there is specific, very nice correlations between the two expressions. This is how we drive the translation. So we do basic research. We put in a lot of patient material. We have a facility for microdissection of patient material, and then we identify therapeutic models, our patients for therapeutic panels. For instance, this one is a new TNBC subtype, for instance. But currently, we're doing a lot about exosomes and CTCs in terms of liquid biopsy. And my last slide is basically, I showed you this. What is the significance and relevance of these amplifications Now we can see that there is, for instance, this particular amplification, which is higher in the invasion front of matched. So we have primary and invasion fronts which are matched, and we do a fold change, and we see that it is increased in the invasion fronts. So the significance and relevance is actually in understanding early invasion, early metastatic disease. So we put together all these panels under the Simpliplex brand, okay, and then they are available for use. And it is a joint venture between University of Malta, Omnigi Medical Technologies, Thermo Fisher and Luminex, and that's the technology that is being set. Thank you very much.
Thank you very much, Prof. I think we'll take just one question, one pending question, and then the others we will reserve for later for Prof.
So one of the hopes for exosomes would be that you can use them for early detection from liquid biopsies, either from patients as a screen, or potential patients as a screen, or from patients who have been treated to see whether they're getting recurrence. Are we any closer to that, or are we still at the point where all of the studies are showing that exosomes from patients with cancer can be identified, but exosomes from patients without cancer identified by any other means, but that go on and develop cancer, as prospective studies, can be used?
So yes, so the problem is that exosomes come from everywhere. Okay, let's set that. So you have to find really the tumor-derived exosomes. Now, the idea of immunoprecipitation is very important, and it's good because those are characteristics of the tumor-derived exosomes. The problem is that we need to know what's on the cell surface markers, same as with CCCs. Now, we are generating, okay, panels of cell surface markers on various types of tumors. Okay? The idea is to merge these antibodies and capture all with different beads, because then we can read them in a multiplex assay, like the Simpliplex assay. Now, in addition to that, we looked at amplifications. So we're looking at amplifications, EMVs, which are more characteristics of tumors. Okay? And we're looking at those that are present in the primary, but not in the metastatic. So going a little bit earlier. Now we're moving even earlier because, for instance, in colorectal cancer, we started a project on the FIT positives that are colonoscopy negative, those that are then early adenomas, late adenomas, and carcinomas. So this is, the idea is to actually find the right characteristics to precipitate those exosomes that are of most interest, and then come in with these huge panels, because we're reading mRNA in exosomes. Okay, very few institutions worldwide are doing that. Mostly they use microRNAs. Okay, and we're finding a lot of sort of specific mRNA in exosomes, which are tumor-derived. So, yeah, hopefully in the next, but we're working on it. At least from my side, I'm working on it.
Thank you very much, Prof. That was very illuminating. Our next speaker is from the New York Genome Center. He is a Dr. Sorry, I'm saying Prof. Sorry, I've got my. He is Dr. Nicholas Robin, who is working. He's specialized in cancer genomics, RNA-seq, single-cell genomics, and currently leads a team of 10 scientists and analysts. He is qualified and he's got a PhD in computational biology, and he also is a part of the Polyethnic 1000 and CZI's Ancestry Network for the Human Cell Atlas, with a project on multi-omics maps of the human endometrium in diverse ancestries. Dr. Nicholas Robin.
Thanks for the introduction, and thanks for inviting me. I apologize for not being present on site with you, but I wish you a good afternoon. And I will present today the project that I'm leading at the New York Genome Center called Polyethnic 1000, which you may have heard about from Melissa Davis earlier today. And I will try to not to repeat myself, but to repeat her talk. But provide a little bit more information. So I joined the New York Genome Center about 11 years ago, really at the formation of this center, which is located in Soho in New York. And I know some of you have visited already, but I invite you to contact me when if you happen to be in New York. I'd be happy to give you a tour. But we were created really by 12 institutions in New York, universities and hospitals, to establish a place for large-scale genomics, human genomics in New York. So we work on 3 disease areas: neurodegenerative, neuropsychiatric disease, and cancer. And as you can see, with a large number of partners in the New York region. So my team is in the computational biology group, and we established a somatic pipeline for whole genome sequencing that used to run on our servers that now can run on the cloud. And I indicate the link here. We're happy to help you install it and work with that if you're interested. But basically, we do alignments of the reads and somatic variant calling. So we detect all type of somatic variants that can happen and that can be detected in the whole genome: single nucleotide, indels, structural variants, copy number variants. We also run a pipeline for germline analysis, which follows the standard GATK pipeline. So we really can identify all type of genetic variation in the patients and in their tumors for follow-up analysis. So as was described by Olivier Elemento earlier today, we apply this pipeline in the NCI Genome Data Analysis Network, and we have run with a number of cancer type and applied our pipeline on those. Out of this pipeline, we also have a pipeline for RNA-seq, and I will describe a project that combines both whole genome and RNA-seq. But we can do a lot of additional analysis on detecting somatic signatures, microsatellite instability, detect HLA subtype, ancestry estimation, and I'll go into detail of that. But as you can see, a number of additional analysis. So the premise of this Polyethnic 1000 project is the fact that public database in cancer genomics are so heavily biased towards patients of European ancestry. So I describe here the pie chart indicates on the left the TCGA, the Cancer Genome Atlas, and on the right the AACR Genie project, and both of them have between 75 and 80% of their patients are white patients from European ancestry. So in contrast, we know that there are some variation in frequency of certain somatic mutation. In particular, here, the example is for lung cancer and a very common mutation in EGFR gene, which we see varies from about 10% of the patients with lung cancer in Africa and in Europe, and goes up to 50% in East Asia, and then has between 10 and 30% in the American continent. So there is a really large amount of variation that is not fully explained, that is not completely due to environment or to diet or to difference in smoking habits, but may have some biological underpinning. So in New York, we are very proud of the large ethnic and racial diversity that is present in the city. I present here a map of New York City. You can see in the middle Manhattan, which is colored as different colors, and they really correspond to different neighborhoods in New York. But you can also see in the north part of the map, the Bronx. In the right side of the map, the Brooklyn and the Queens. But I also represented New Jersey, and you can see that the color correspond to the census. So the regular annual or every 10 year, the U.S. government goes and try to ask a number of questions to study demographic in the entire country. And here we can represent the ethnic diversity of New York, but also represent and acknowledge the large segregation between those different neighborhoods. So we decided to take advantage of this great diversity and really identify or try to study cancer genomics in patients of different ancestry, knowing that it will be a very, very small step toward a complete understanding of cancer in different populations. But that's something that we can do locally in New York because we think that we have represented from large part of the world. So we established a pilot phase that we call phase 1 in 2019, in which we collected 160 tumor samples, archival samples from 13 institutions in New York. All of them were self-identified as non-white. So that may be unfamiliar to some of you, but when patients go to the hospital in the US, they have to identify a questionnaire, and they have to identify, or they can decline to identify, but most patients identify themselves as white, African-American, Latino, Asians, or various other categories. And so here we only took samples that were non-white. We perform whole exome and RNA sequencing on the tumor, and we run a tool for genetic ancestry estimation. And that's represented here on the right side of this slide, where I represent the cell declaration at the top, and then the continental level, the quantitative estimation of the continental ancestry, and further down the population level. So using the 1,000 genome reference population, we can identify and quantify the ancestry or the genetic ancestry of those patients. We can do that directly from the tumor, and we see that even with patients that are self-identify as African American, there is a variable amount of genetic ancestry coming from the African continent, and within those, there is a large disparity or differences between the different ethnic origin within the African continent. And really, we think that's the level of differences that we are going to be studying in this project. So you can see here we have African American patients, we have Latino patients, and we have East Asian samples. So we published an essay that described this first phase of the project that came out last year, and then we started the second phase of our project. So for which we assembled a review board that helped us to identify the right project, and we assembled a budget for tumor normal whole genome sequencing, tumor RNA-seq, and about 1,000 retrospective and prospective samples. And all of those samples were consented for research and for broad data sharing. So the first outcome of this project will be to share the data in public database. And we have support from various philanthropic donors, industry donors, and institutional support. So we started 7 different projects on bladder cancer, breast and prostate cancer, pancreatic cancer, multiple myeloma, lung cancer, colon cancer, endometrial cancer with different institutions. And you have here snapshots of the different PI of those projects, and you can identify Melissa Davis and Onye Balogun. Both of them are at Weill Cornell and are affiliated to this project as ethnicity and cancer scholars for the New York Genome Center. So far, we have received 600 samples and have sequenced 507 of them. And that's the results that I can start to present to you today. So the genetic ancestry estimation is represented here, separated by the different projects. So on the left, we have the first project is the bladder cancer project, where we have a few samples. Then the breast cancer project led by Melissa Davis, followed by the colon cancer, multiple myeloma, or lung cancer, sorry, multiple myeloma, and pancreatic cancer. And so you can see that in 5 of those projects, we have a large proportion of patients from African American or with African ancestry. The exception being the lung cancer project in the middle, which is designed as a project to study East Asians and European in New York for early cancer. So the top track shows the continental level ancestry, and the bottom track shows the population level ancestry. And so you see that there is already a very large amount of genetic diversity in this project that will really be much larger than what anything has been sequenced before, and those samples are going to be shared in public database. So what we can do with the somatic pipeline that I described is to take into account all of the somatic SNVs and decompose them into the mutational signatures using the Cosmic Reference Database. And that's shown here. So again, we have the first project is bladder, breast cancer, colon cancer, lung cancer, multiple myeloma, and prostate cancer. And I'm not going to go into details, but all of those representation are the bottom track shows the single base nucleotide, the SNVs, then the doublet base substitution, and at the top, the insertion and deletion. And all of those all of those bars correspond to different mutational processes. Some of them have been identified and associated with processes such as smoking or ApoBec mutations or different chemotherapy. All of those samples, all of those plots here are sorted by the red bars at the bottom, corresponding to SBS1, which is the canonical signature associated with aging. And so we see that we have various proportion of those somatic variants are associated with aging, but we also have in the lung cancer, we have a number of samples with those orange bar corresponding to SBS4, which is known to be corresponding to smoking signatures. In all of those different cancer types, we identify some of the known and expected mutational processes that are what makes all of those cancer types different because they're exposed to different mutagen or they really developed through a different mechanism. So those are a confirmation of the processes that we expect to see. Thanks. So I would just want to go very quickly a little bit more detail on the project that for which the sample collection is completed, and that's the lung cancer, which really studies early lung cancer in African American, East Asians, and in European patients in New York. The somatic signatures are represented here, and you can see here if we separate on the left the Asian patients and on the right the white patients. We can identify the known signature associated with smoking, this SBS4 that I talked about, as well as the DBS2, and you can see it in both of the cohort. You can also see when we look at the information about those patients that we have gender information and smoking status information, and unfortunately those two things are quite confounded in the sense that most smokers in our cohort are male. And that's true for both white patients and Asian patients. But there are multiple other information that we can extract and we can try to analyze in those type of results. If we go to our examples of EGFR mutation, as I described earlier, we know that that's a canonical example of a large amount of diversity in the different population. And in our small cohort, we identify EGFR mutation in 62% of the Asian patient, and only in 41% of the white population in this cohort. So already we see a difference, and the type of variance that we identify is quite different. So that requires further analysis. If we look at the mutation burden between those two groups, we don't see a statistically significant difference. And so we have the RNA-seq, and we also look at the expression profile. We can do a differential expression analysis of those two populations and try to identify, what are the differences, what are the groups of genes that are overexpressed or underexpressed in one population versus the other, and try to correlate that with potential differences in the outcome. So what we are doing now is to complete the samples collection, to start sharing metadata and clinical information, have a regular discussion with all of those different individual teams, start to publish some papers coming out of this project, and really will deposit all of the data in public database such as ICGC Argo and potentially others. There are starting to be some posters that are presented. You can see here Melissa Davis and Onye Balogun presented our project at the AACR Health Disparity Conference in September. And in the future, we hope to continue this project, expand the existing cohort, add additional cancer type, add more population, and do a large effort in patient community outreach. And obviously, we would love to have corresponding effort in the different countries so that we can compare, for instance, our African American cohort with African cohort in different countries. So we know it's a very small effort to address the large problem of cancer health disparity. And here it is summarized, the problem is summarized in this report from AACR, but we know we can sequence and analyze genomes and RNA, but we're open to discussion about what else we should be doing, with whom and for whom. So with that, I want to thank the people in my team that play a large role in analyzing this data, the project management, the leadership from the New York Genome Center, the ethnicity and cancer scholars Onye Balogun and Melissa Davis, our Polyethnic 1000 steering committee led by Harold Varmus, and all of the support that we receive for this project. I want to thank you very much for your attention, and I'm happy to answer any question.
Thank you very much again, Dr. Robin. It looks like we're still formulating the questions to be posted to you. We thank you very much for your time. Thank you.
Thank you. Bye.
Our next speaker is Prof. Bida. He's going to give a keynote address on precision oncology, bridging the translational gap. Prof. Bida has worked in several institutions, both as a chief specialist and as a professor in various institutions. He is also a Pacrinian, if I were to say that. He is going to be sharing his insights in terms of how we translate all this the genomic space as also the information. How do we bridge the gap between the discoveries and also taking them to the clinic? Thank you very much, Prof. Bida.
Well, thank you for that introduction. I'm just going to share with you some insights in terms of how we can increase the transformative change that we see quite often in molecular genetic analysis. And we do know that nowadays we can carry out wonderful things like whole genomic sequencing, study for the whole exome sequencing, but we generate large volumes of data. And what we have, compare what we have at hand with what we actually can use on the clinical platform, we find that there's a disparity there. So there is a great gap in terms of translation. Most of this can be seen in the areas where we are doing polygenetic analysis, because most of the monogenic cancers and diseases are quite well established because we've got candidate genes that are also available. But for polygenic analysis, we can still glean a lot more information that we can transform into clinical practice. This is in the area of risk analysis, for example, in the area of predictive prognosis, in the area of therapeutic responses. But when we look at a number of genetic changes in any cancer sample, we can then construct ways and means that we can deploy that information into clinical use in terms of what we do. We do know that this translational gap is actually caused by the fact that there is now a lot more information we can generate, but little of what we can translate into clinical practice, as I said earlier on. Somatic mutations are really what characterizes cancer in terms of cancer biology, but what we need to look at now is what are those somatic mutations that are tumor-defining molecular events that we can actually utilize fruitfully in terms of a clinical platform. Now, throughout the decades, we have been dealing with quite a lot of monogenic analysis where candidate genes are well-defined and catalogs are available, but we now have to look at a broader scope of implementation that goes beyond that level of analysis. When we look at the relationship between translational medicine and precision medicine, we see that in the earlier stages, what we did was to construct disease models which closely mimic real situations in the clinic, and therefore deduce whatever we want to do and design it in such a way that it recapitulates and mimics the real life situation in the clinical platform. Most of that has now been largely replaced by biobanks who are able to generate tumor samples and study them right from through patients, and that actually has increased most of the transformative ability of the information. We then deploy multi-omics, we deploy biomarkers, genetic biomarkers, and those can then be taken directly from the experiment into the clinical platform because of the nature of the time that we use. And we then end up with a disease endotype with large volumes of information that in fact we use quite a little bit of that, not enough in terms of what we have at hand. We look at the clinical trials and see how they interface with multi-omics, and we can also see that interventional clinical trials are still lagging behind because that really results into patient response, a real life patient response, dynamic changes that occur in the patient, whether it is in the in the blood, whether it is in the organs, whatever, then we need to also marry this also into real life situations. The interventional clinical trials are still lagging behind in terms of the transformability of the molecular information we get. But then we have multi-omics, which is actually an area where there's a lot of information we now can generate with defining endotypes of diseases, and we can then marry that into the biomarker analysis, and eventually we can also increase that transformative ability by implementing what you call companion diagnostic, which helps us in terms of clinical trial design. And then we end up with what you call precision medicine. So there's quite a lot of steps that are involved, but however, in each of these steps, we have quite a lot of things we can do in terms of improving that transformative ability of the data that we generate. So in terms of the omics technologies, that has been a single most important revolutionary step in terms of precision medicine, where we can see that the amount of data we have must be actually integrated to a point where we're able to know what's happening in terms of the nuclear properties of the cancer cells, cytoplasmic changes, and everything else. Together, we integrate all that into a conceptual understanding of what is happening in the cell, and therefore able to utilize that information with risk prediction and other aspects of it. We also know that most of what we have in terms of the experimental design, some of it tends to be false positives and false negatives, and therefore we have to have controls. But when we carry out experiments in multicentric situation, controls may not be standardized, and we have an issue in terms of what we find to incorporate that information into patient care and clinical decision-making process. Now, polygenic analysis has now become the buzz thing in terms of molecular genetics, because we end up with a lot of genetic alterations that occur when we've got small tumor samples. We then generate all this information, but what can we do with this information? One of the areas where we can deploy this polygenic analysis is to deploy them in the risk assessment. Risk assessment in terms of occurrence of the disease, risk assessment in terms of recurrence of the disease, in terms of therapeutic responses. Now, all that information is very vital in terms of how we can utilize it and improve our translational ability of our data. So polygenic analysis has been used in some of the parts of the world where they used it in terms of risk analysis using the concept of polygenic risk scores. Now, polygenic risk scores is a combination of molecular data that is generated that can be used in terms of analyzing the risk of occurrence of the disease in the preventative medicine in terms of primary health care as well. Polygenic risk scores can be done, generated, but they need to be carefully used, because there is also the issue that genetic expression can often be influenced by environment. So this interface between genetic expression and environmental influences remains a very important aspect of actually quantifying the risk of occurrence of disease using polygenic risk scores. Then after that, we can also look at things like what other things come into the picture. We're not actually talking about the ancestry genetic susceptibility that is well defined in most of the cancer types, actually based on things like germline mutations and other known candidate genes. We're talking about a situation where polygenic analysis data is available. We can use that in terms of risk assessment and actually in terms of analyzing which patients are much more at risk, and therefore they deserve to be subjected to surveillance programs of routine screening. When we've taken out these ancestry genetic changes, we are now left with what you call polygenic risk scores, which can be incorporated in our data sets in order to manage these patients. There are three methods that we can use in terms of utilizing polygenic risk scores. We can use what you call percentile ranks, which is nothing else but the bell curve analysis of what you have when you have comparing that with the actual population that you're choosing. If we're choosing breast cancer patients, a series of genetic alterations, taking out, for example, things like BRCA1 and BRCA2, these are well-known candidate genes, but talking about general population where cancer arises in a patient in the background of absence of these kind of candidate genes. In other words, if we look at the statistics, about 20% or so of women will have BRCA1 gene, so majority of them won't have that. But we still need to know if we can implement polygenic analysis in terms of assessing risk of this patient developing breast cancer. So we can use percentile ranking. The second method we can use is what you call relative risk assessment and absolute risk assessment. And the percentile ranking is a little bit much more less accurate because it doesn't exclude, as I said earlier on, non-genetic influences that may come into the picture in order to assess the risk. But when you look at a relative effect of this, we can look at define the average risk that exists in that particular population at risk. For example, breast cancer patients, we know that if you've got this set of data sets in terms of genetic alterations, these patients are known to have breast cancer. So therefore, the patient may present with a higher level of average risk, and that is the way we can actually assess the risk of occurrence. The much more, perhaps closer to accurate way of prediction is what we call absolute risk. And absolute risk usually define the absolute risk which is based on genetic influences with minimal interference from the other environmental factors. So that is what we can do in terms of assessing the general population that are actually seeking to be screened for breast cancer. Then we end up with what you call a threshold which is defined based on one of the three methods, defines the level at which the patient, if the patient scores higher than that, the patient therefore will have to be undergoing regular screening programs such as mammograms and amongst other things. And it's interesting that these patients will often pick up precancerous lesions, and therefore that is actually a very important step because we can actually prevent cancer if we pick up these precancerous lesions because the patient has a higher score of a PRS score, other than just using manual method of breast palpation, because breast cancer, as we know, like any other cancer, starts off as a single cell, then 2 cells and 3 cells. And by doing this, we're able to therefore subject the patients to a much more frequent evaluation in terms of a screening program. Now, of these 3 methods, it's important to also remember that most of them, like the 2 methods are defined, they are not really excluding the non-genetic influences. The one that stands out is the one that I said is the absolute risk, which is very important in terms of implementing in a clinical situation, in terms of trying to monitor our patients. So we got the 3 options, and as I said, and these options are very important, especially when you're dealing with a particular type of cancers. Breast cancer is one of them. In some other cases, we can also apply the same principle when you talk about colorectal cancers, and then other cancers that are subject to these polygenic alterations can also come on board, and we're able to narrow the scope of actually knowing which patients must be subjected to particular screening programs. And then that is what is important in terms of risk occurrence. Now, when we look at the general application of this concept, there has been a few examples on the market, such as Ambry Genetics, which has now been used in prostate cancers as well. And we also have the Tyrer-Cuzick method model of genetic screening, which has been used in breast cancer. So these are some of the tools that have been used in terms of screening the risk of occurrence, other than relying on the currently available method, because by the time you pick up something on a mammogram, quite often it's too late. The cancer has actually doubled up, if not gone to regional lymph node basin. So those are some of the things that we can do. In terms of the breast cancer in particular, we have what is called early breast cancer monitoring in terms of clinical application. The early breast cancer monitoring is what happens when you have actually removed the breast cancer, and it's an early stage cancer. You want to detect whether the patient is at risk of having a breast recurrence. Now, we're moving now to the step of recurrence, using polygenic alterations in recurrence. So in terms of breast cancer, when you've done surgery in stage 1, stage 2 breast cancers, and you want to know whether the patient must be subjected to adjuvant therapy, these are some of the important clinical decisions that you can make based on the PRS scores as well, and based on the other commercial available kits that are out there. One of them is PAM50, MammaPrint. They use the same concept of a group of genetic alterations. They are ranked in particular pattern, and they're able to provide and quantify the level of risk, and therefore one is able to pinpoint which patients are important in terms of following them up closely and look at the the preventative aspect of recurrence in this particular patient. Some of them are so well developed that they actually can tell you whether it's going to be an early recurrence or a late recurrence. So those are important clinical decisions to be made, because sometimes the patients may actually be subjected to adjuvant therapy for a period of time, and you stop the treatment because you have found out from these analyses that the patient is likely to have an early recurrence, other than giving the patient a long-term protracted treatment and therefore subjecting the patient to a number of side effects in terms of clinical treatment of these patients. What about in predictive prognosis? And then one of the important aspects we know about cancers is that the concept of tumor burden, tumor mutational burden, is very important in terms of defining prognosis of cancers. Now, tumor mutational burden is almost like what you can say is an index of proliferation of cancers that we sometimes use in static light microscopy, assessing using Ki-67 amongst other markers to detect how aggressively dividing the tumor is, and therefore those tumors require a particular dosage in terms of chemotherapy, and therefore you can adjust your treatment based on the tumor mutational burden. However, the accuracy of this technique requires to be refined, because tumor mutational burden sometimes is looking at a number of kinds of components in a cancer. Cancer has a number of components in it. For example, you can have a cancer stem cells, you can have a cancer phenotype that is in the cycling phase of the cell cycle. You can have other cancers that are cancer cells that are in fact on their way out in terms of apoptosis, they're senescent cancer cells. So when we talk about this, the general concept of tumor mutational burden doesn't actually, we then have to purify this concept, and so that we can use what you call corrected tumor mutational burden. That is a much more precise way of actually assessing the proliferative index of that particular cancer. So a corrected tumor mutational burden is a concept which has now come up, which actually identifies, which of the cancer types that are undergoing treatment would require dosage adjustment, would require an additional drug to be added in a multiple dosage treatment of a particular cancer. So that's an important concept in terms of treating the patients in a clinical platform. So we also know that when we have a corrected tumor mutational burden, we have a much more precise tool that can actually be used. This is a concept where you're only taking into account those cancer cells that have undergone mutation, because you want to target them using chemotherapy and be able to shrink the cancer. And therefore, that is how we can apply this concept in terms of applying in breast cancer. The next thing is about the non-coding driver mutations in cancer susceptibility. Driver mutations have actually been around us for a long time. They actually are best understood, best known in the coding regions, but the non-coding regions, in fact, can sometimes harbor particular mutations that in fact can act as driver mutations. So the way to look at it is that when we do exon analysis without doing whole genome analysis, we end up with only what you call driver mutations, because they are coming from the coding regions of the DNA segment. So expanding that through the assays of whole genome analysis will reveal some of the genetic sequences that are present in the cancer, that giving rise to a definition of this type of changes in the cancer that we're able to identify and target in terms of a diagnosis. So this is an example of how we can actually identify the cancer, the non-coding driver mutation. We use three methods. You can use what you call annotation-based, use what you call mutation rate-based, we also call correlation-based. All of these are linked to analyzing and scanning the whole genome and be able to identify. For example, when you talk about annotation-based, we are looking at areas where there is a polymorphism using particular annotations like the ETF that actually identifies the areas of the DNA segment that are in fact areas of mutation in the non-coding region. Rate-based is based on the rate of mutation, where you can identify hotspots using the whole genome analysis as well. And then correlation based, you correlate these with the areas that are actually actively dividing using the RNA sequencing that in fact can actually generate that. So we define the hotspots using the 3 methods, and then eventually we are able to identify which areas of the non-coding regions in fact can end up as being a driver mutation, and you analyze that data into the whole data set that you have. And the next is what you call single nucleotide polymorphism. And this is a variation of the DNA sequence that often sometimes occurs in the general population, but when it occurs in cancer, that can give rise to false information. It can give rise to difficulties in identifying particular cancer types, whether it be in terms of diagnosis, in terms of response to treatment, that can provide quite a lot of challenges. Therefore, through whole genome analysis, we're able to define these other DNA segments in terms of what they are, in terms of giving rise to what they can do in the cancer type. Then we also have what you call response to therapy. When we talk about response to therapy, we look at how the tumors will respond to particular targets and particular chemotherapy. And one of the good examples is the Tata box, where if you've got a mutation, a single nucleotide mutation, you can actually have a problem in promoter sequence activity that actually gives rise to cancer. And whether you are doing it with epigenetic, whether it's introns or exons, the SNPs can actually play a major role in terms of confounding the result. Then the last thing is about the heterogeneity of tumor. Intratumoral heterogeneity, whether it's based on the space or temporal heterogeneity, what we use as a simple, small tumor samples is not a complete representation of what is happening in the whole of the tumor. So we need also to adopt methods that can actually help us to analyze most of the tumor samples using multi-region sampling techniques and to define multiple areas of tumor that can be responsible for cancer dynamics. And this is an example of areas where you can see multiple regions of the tumors. In the case of lung cancer, we can see they are very variable. And if we target one small gene there, we are going to be at a loss in terms of analyzing what it is. And then we also put it in what you call heat maps that generate areas that we can map out in a cancer. So that is more or less what I have for you. And in conclusion, I will say that most of what we have is not what we can use. So we need to improve on what we have so that we can implement more of what we have in terms of cancer dynamics. Thank you very much.
Thank you very much, Prof. Bida. He moved to the Netherlands Cancer Institute for his PhD in cancer biology. And during his postdoc years at the Craig Manchester Cancer Institute, he studied the role of specialized lipids called phosphorinositides in cancer cell migration. And at this point, lentiviral delivery of shRNA as libraries had become available, and they used this together the newly developed high-throughput migration assay. Currently, Prof. van der Bout heads a cancer biology group with a focus on the role of hormone receptor GPCR proteins in breast cancer. To this end, they are also establishing a breast cancer biobank as their test platform, and they are testing a novel cancer diagnostic tool which uses ultra-low pass whole genome sequencing to analyze cell-free DNA isolated from blood to detect cancer-specific DNA. Thank you very much, Prof. Uman.
So without further ado, as has already been discussed to quite some extent, there are definite differences in cancer in different populations, and especially when we compare African populations to the European populations. But as also has already been said, there is a clear lack and a clear bias towards cancer research in European populations. And that has a historical background, of course, but it also is part of the other reason is also because lack of access to patient samples and lack of infrastructure within Africa to actually be able to analyze these. And I think we can all agree that some of the previous talks were absolutely excellent, but they are like they're on a different planet right now from what we are able to do here. At the bottom there, you can just see a good example of why we should be focusing on Africa-centric research, cancer research, because you can see that there are differences in some important cancer genes in terms of their frequency. So some of them, while being very prevalent globally, are not prevalent within African populations, and the opposite is true, that certain mutations are very prevalent in African populations, but not so in European or in global populations. So I'm within the Department of Physiology. I'm an associate professor there, and I work within the Center for Neuroendocrinology, which is a big word that we don't do too much about, but our focus really has always been on GPCR receptors of hormones. And within that center, we've set up a cancer group, and that had looking at hormone-dependent cancers as part of its remit. The mission of our group is to establish better diagnostics and improve treatments for South African patients specifically, and we do that by establishing novel molecular diagnostic modalities. So we're looking at analysis of cell-free DNA as one option in order to improve ease of access to diagnosis, and also to hasten the path between diagnosis and treatment. And then what I'm going to talk to you about today is to establish a research platform making use of cancer organoids in order for people to study either cancer biology, genetic and proteomic factors, and also for things like drug development. And then lastly, I also have an interest in GPCR hormone receptors and their role in cancer biology. So for this talk, we're going to look at this establishing a platform for preclinical breast cancer research, and that is to establish a living biobank by isolating cancer cells from Black African patients specifically and establishing cancer organoids. And with these, in our project, we're actually assessing the effect of chemotherapies that are currently used in the public health, trying to identify good and poor responders, and then using proteomics and genomics to try to collect a marker profile that we could then use in an immunohistochemistry setting or in a basic genomic setting to actually start to stratify patients. So we have established a small panel of lines, and we're busy doing chemotherapy response, and then we do proteomics and genomics to understand what is happening. Now, it has been said before, but COVID has had a massive impact, especially if you work within the public health system, because we had prolonged, and with prolonged I mean months-long closure of the slate, which meant that because breast surgery is elective, there was no breast surgery in the last 2 years. Almost half of that time, we didn't have mastectomies, especially on naive patients. So there has been a big switch in treatment where a lot of patients got neoadjuvant treatment instead of first being resected and then getting adjuvant treatment. That's why we haven't been able to collect as many samples as we would like, but nonetheless. So just for those that don't know what organoids are, and there's no shame in not knowing that, so organoids is a system developed by Hans Clevers in the Ubrecht Institute in the Netherlands, where you can take cells from an organ and you can isolate them, and by growing them in a matrix gel containing different factors, including stem cell factors, etc., you can ensure that the stem cells remain alive, they proliferate and self-renew, and they also differentiate, and their progenitors differentiate to form, in essence, the base of that organ in the Petri dish. And the genetics of what is in the Petri dish here is extremely similar to what is in the actual organ. Now, this can be applied to cancer as well, and remember that cancer is a heterogeneous disease. So within one tumor, you have many subpopulations, and the problem with classical cell lines is that we actually select out many of those populations. So with cell lines, you have a very homogeneous population, but what we want is we want that heterogeneity, because remember, when you treat cancers, what you get remission for is usually a small subpopulation that has picked up resistance to whatever therapy you gave. So for cancer organoids, what we do is we take a small sample of the resected tumor. We can then establish an organoid line. And this organoid line, what is really handy about it, and this is why this is a platform that can be used by others, is it's expandable, so we can grow lots of it, but it's also storable and revivable, so that even if 10 years down the line you want to use it, we can revive these and reuse these. The other thing that's really important, of course, is this is now a living system that we can test drug response on, rather than depending on just patient samples of dead cells in essence. So how this actually works in our project, we have collaborations with 2 surgeons, Malosi Makgoka at Steve Biko and Brandon Jackson at Kalafong Provincial Tertiary Hospital. And when they get a patient that they're gonna perform a mastectomy on, we get called and we go and collect patient data. We collect tumor tissue and we collect some blood. From the blood, we'll do genomic DNA extraction for analysis. The tumor tissue gets divided, and part of it is used for genomic DNA extraction, and the other part will be used for organoid establishment. And this is what that looks like in real life. So we go to the theater and get a little piece of the tumor. This tumor is then digested using collagenase, so that we end up with a palette of cells that not only contain tumor cells, of course, as you can see, there's red blood cells in there as well, and the fibroblasts, etc. But then we grow them in a dish with dots of matrigel, and the factors that are in the media will specifically allow the tumor cells to remain alive, while all the other cells will eventually die. So after a few passages, you will have a pure tumor population. And this is what that looks like. So straight after isolation, you have these small clumps of cancer cells. They're easy to recognize because normal cells and red blood cells are much smaller than these cancer cells. And as you can see over time, these grow nicely. And if you were to leave them for very long, they will differentiate in different shapes, and they will even start to grow out and migrate away from the tumor. So you can really recapitulate an in vivo tumor environment in this way. So far, we've collected about 14 samples, 5 were lost. Again, this is technology that I only know of one person that is doing cancer organoids in this country. It is new, so we have to learn. So we lose a few, but nonetheless, we've established 9, and this is the different pathologies that we have. As you see, not many triple negatives because they're quite rare. Most of them are luminal A. So what we want to do, of course, is to test how they respond to the chemotherapies that are commonly given within the public health system. Remember, we're only focused on that. And within the public health system, the sort of adjuvant treatment one gets is adriamycin or doxorubicin, cyclophosphamide, or some of the taxanes like paclitaxel. Of course, there's also hormone treatment, which is basically tamoxifen, and in some cases Herceptin if you're lucky. But that's about the 5 treatments we have, and here we're just focusing on the chemotherapy. On the left here, we've used a cell line, it's a triple negative cell line in fact, called BT20 to set up our assay. And you can see over time a nice growth. So these are grown in the same media as organoids and also grown in 3D. And you see a nice growth curve over time, but if you treat them with doxorubicin, you have no growth at all. On the right-hand side is preliminary data. It's 2 repeats, and it doesn't look like a sigmoidal curve at all. So here's a log curve on the X scale, and this is for both cyclophosphamide and paclitaxel. And you see that we don't really get a drug response for either of these drugs in this specific line called 6767. So that tells us that it's possible that this line is resistant to those 2 chemotherapies. So in that way, you can already see how this can be really interesting, because now we can do the genomics and proteomics to start to understand what markers are there that could predict this for us from a tumor biopsy. So what we do with that, and this is, there's no data on this. I have list of SNPs, but we haven't analyzed it yet. So we're using a 50 cancer gene panel from Cellomics, and with Incaba, we're sequencing both the tumor, the germline, which is represented by the blood sample, of course, and then the organoid, for two reasons. One is to identify all of the variants that are present in order to identify any novel variants, but also known variants of these 50 cancer genes in the sample. And the second part is we can use this panel to determine the similarity between the original tumor sample and the organoid line as a way to do QC on our organoid line, so that we know whether we are losing some of the subpopulations that were originally in the tumor. We also sent these samples away for proteomics. Again, this is occurring as a way to set up this pipeline, because now we need to do a mass spec for breast cancer specifically. So what we did is we again took the BT20 cells and we grew them in 3 different formats. So either as 3D spheroids, so normal media, just losing all attachments, so they have to attach to each other. In 2D, which is the original format, and then also as organoids, which is also in 3D, but then with this specialized media. And we did mass spec to try to see how sensitive our method is to identify differences in protein expression. And you can see nicely from this Venn diagram, you get some unique proteins that are expressed in each of these formats. And if you look at the PCA plot, you can see very nicely that they separate out quite far away from each other, which means that we are getting profiles of protein signatures that really allow us to identify specific, in this case, specific formats, but this will allow us to identify specific organoids lines as well. What we're also doing is, as a cell biologist, I'm always interested in what cells look like and where proteins are. So we use confocal microscopy to just look at the morphology of cells, of how they behave. And these are just some examples. This is cadherin, actin, and beta-catenin staining of organoids. And as you can see, these form very nice cell-cell contacts that both have actin around them and then beta-catenin and cadherin. So here I want to just show you an example of what I'd like to call a good indication that we are on the right track here. And that is a organoid line that seems to be behaving quite similarly to what the tumor is behaving like in vivo. So this patient, 6767, presented initially with a small tumor, but by the time of surgery it was a stage 4 a T4M0 tumor, so very large tumor, only local invasion, very smooth tumors as well. Whereas the 5733 was a T2M1, so this was already metastatic, even though it was only at the T2 stage. So these are very different types of organoids. And what we noticed when we cultured 6767 is that some of the cells would escape the matrix gel, but they would never attach to the plastic, whereas normally all of these cells will attach to plastic and grow in a 2D fashion. And that's what we see here. So here on the left, you have 5733 cells grown in 2D, and you can see nice flat cells. They've attached to plastic and they're now growing in 2D. In contrast, the 6767 won't do that. And even without matrix gel on plastic, they will not attach to it and they'll still form organoids. So clearly this cell line is missing a very important cell adhesion protein. We did adhesion assays here on BME, so this is the best position you can get for cells to adhere to, and you see that we get no adhesion at all, in contrast to the 5733 line where we do get adhesion. So this clearly tells us that in vivo we get this massive tumor but no metastasis. It forms very smooth, it stays in its primary site, and in vitro we see similarly that they don't adhere, which means that they don't have the capacity to attach to and move along stroma, which is a basic premise for metastasis. So what we're doing now is we're doing all the growth curves to check for sensitivity of all the lines. The genomic and proteomic analysis is ongoing right now. And from that, we hope to identify markers that we then can then go on and test in patients to see if we can stratify according to these lines for a drug response. And then, of course, the 6767 line is very interesting, and we're very interested in why there's such a complete lack of adhesion. Lastly, I just want to thank, first of all, the funders for this project, because growing organoids is not cheap, so the MRC provided SHIP funding. Then the research team is the two surgeons, Drs. Malosi Makhoka and Brandon Jackson. Cindy Seaway and Pravin are at the CSIR, where they do the proteomics. And Richard is part of the Department of Oncology and helps me with follow-up of patients. Adela is an MSc student that works on this. All the patients that take part, the Center for Neuroendocrinology and the two hospitals, really importantly. And then lastly, I want to bump, I'm treasurer for this society. It's a Society for Advanced Cell Culture Modeling. And we have a conference in October where you're absolutely welcome to attend. It's a really great conference. It's in Potchefstroom. So you can't leave, you go there and you stay. But if you want to know more about advanced cell culture models like organoids, this is where you want to be going. Thank you very much for your time.
Thank you very much, Prof. Do we have a question?
So I think, please correct me if I'm wrong. I think the question is should I have looked at an earlier process and also, I think you thought I said there are no adhesive proteins. That's not quite correct. We don't know what's going on. So it's difficult to understand why you would lose complete adhesion. So they clearly can adhere to one another. So they have cell-to-cell adhesion, but they don't have cell-substrate adhesion, which is integrin mediated. But they will probably have multiple integrins, so they should be able to adhere to at least some substrate, and they don't. So the thinking right now is that it's actually more a cytoplasmic protein, something like talin, that regulates integrins that might be mutated or no longer expressed that is doing this. And so we can look for that, and we are busy looking for that. Unfortunately, we haven't been able to get good stainings to look under the microscope for it. But that is definitely something that we can do.
Since you're not saying they're not there. Oh, sorry. What you're saying is the adhesion proteins are not necessarily not there. You're just not sure why the C6767 line is not attaching.
Yes. So my bet would be that the integrins will be there, but they're not in their active in their high affinity form, because there are many different integrins. So it would be difficult to imagine a scenario where all of their expression is gone. It's much more likely that it's something cytoplasmic that regulates their activity that is gone. And so, for instance, talin is a good example. Vinculin is another example. So these are single proteins that will regulate all integrins. Yeah. Thank you.
It's also in addition to this one, just out of interest, you mentioned that the 6767 lacks adhesive properties. So if ever it lacks adhesive properties and you're trying to determine which important adhesive molecules it lacks, how are you going to determine which adhesive molecules it lacks, and how are you going to be sure that those are the ones that are lacking for it to attach?
That's a good question, and that is that also leads in why it's so important that we're doing proteomics and genomics, because we need to annotate these lines so that if you want to use it, you get all of that information as well. So proteomic analysis should be able to tell us, especially if we do comparative proteomics with other lines, which proteins are missing, if they are indeed missing, and genomics will tell us whether any of these proteins are mutated. So luckily the adhesive sort of protein family, it's big, but it's not, it's not unimaginable. It's about 250 proteins that are involved in focal adhesions and adhesions. So it's a defined group that we can look at. So hopefully it's one of them. If it's not, then we can still look at things like transcription factors that regulate expression, etc. But that's why we're doing both the genomics and the proteomics, especially because we can find things like this out.
Our next speaker being Professor Lisecha. He's a molecular oncology and apoptosis specialist. He also was appointed as an advisory member to serve on the Department of Monitoring and Evaluation on Health in 2010. He also serves on the American Association for Cancer Research International Committee, representing Africa on how to advance cancer research in Africa. We're going to have Zodwa presenting the slides on behalf of Prof. Lisecha.
Good day, everyone. It's always weird to speak through a microphone, but it's fine. I am Zodwa from the University of Johannesburg. I will be standing in for Professor Lisecha Raymond Mutadi, and my talk today is about CBD, cannabidiol, and cannabis sativa as potential treatments in vitro prostate cancer cells silenced with RBBP6 in prostate cancer xenograft. Now, as we all may know, prostate cancer is classified as the second death-leading cancer type in males aged 50 and above. This is mainly due to its inability to be diagnosed at an early stage. And in 2020, it was found to account for about 14% of the male malignancies, and it was ranked as part of the top 5 leading cancers in 2020. Now, as we all may know, prostate cancer is the cancer of the prostate. As you can see, apologies for that. As you can see, it has side symptoms such as compressing the urethra, and it has also been found to cause a condition such as metastatic spinal cord compression. Its main cause is currently not known, hence the importance for males to regularly screen. It has risk factors such as age, family history, lifestyle factors, and one that is the weirdest of them all, race. Now, if you could look at this figure over here, it shows the prostate cancer incidence and mortality rate. It has been found to be more common and deadly in males of African ancestry. Now, as you can look at the figure over here, it shows that the European and Asian countries tend to have more cases, more incidence cases as compared to the death or mortality rates. One of the reasons behind this, it has been stated that this can be due to African countries being less developed and with individuals which are less informed with this type of condition. Now, looking at this, it is basically the summary of the cell proliferation. It's a summary of the cancer cell proliferation. As you can see over there, the damage of the DNA has been found to result in the production of P53, which is supposed to result in cell cycle arrest and apoptosis. However, it has the ability, cancer progression has the ability to inhibit this P53 further result in RBBP6, and this will also result in the degradation of the P53, which will result in cell proliferation, and hence the need to look into other avenues, such as looking for alternative therapies from medicinal plants. Medicinal plants have been found to be a common research focus due to their unlimited bioactive compounds, and these bioactive compounds have been found to produce fewer side effects. Now, this study was looking into a well-known medicinal plant, cannabis sativa, which has been found to have a compound such as cannabidiol, which has been found to have properties of inhibiting the cancer cell growth. Now, the aim of the study was to investigate the cell proliferation or progression inhibition effect of cannabis sativa and CBD on prostate cancer. And we had hypothesized that cannabis sativa and CBD have a potential cell progression inhibition properties in prostate cancer. And these are the objectives of the study. Moving right along to the methodology, this study had two parts. We extracted cannabis sativa, and then the first part was to culture cells, transfect them with RBBP6 silencer. And the second part, we're working in collaboration with the Northwest University in preparation of the xenograft prostate cancer. And the methods we used in the study include Excelligence, where we wanted to see the real-time cell viability, followed by caspase activity, where we wanted to see if indeed the cells were committing to apoptosis or not after treatment. We also employed MTT assay to also measure the cell viability, and lastly, gene expression analysis and the Western blot. These are just the summary. This is just the summary of the first part of the study, where we used cell culture. And then this is the second part of the study, where we prepared the xenograft prostate cancer mice. Now, moving right along, the results further indicate that CBD alone was able to inhibit the proliferation of prostate cancer. And when we compare it with the crude extract of cannabis sativa, as you can see there, CBD was found to be more effective as compared to the crude extracts of cannabis sativa. This can mainly be to the fact that cannabis sativa has been found to have different compounds which have an antagonistic property, that is, they are able to inhibit one another from whatever function that they are meant to do. And then looking at these figures over here, we also did Excelligence for normal cells, and we also did Excelligence for cells silenced with RBPP6, and we also used cisplatin as our positive control. Now, looking at this figure over here, it shows the caspase activity amongst the different treatment groups, and we're able to see that it's CBD over here. We're able to see that CBD, as compared to cannabis alone, it has a higher caspase activity. And then looking at the CBD group silenced with RBBP6, we can also see that they have a higher caspase activity, and also cisplatin and cisplatin alone. Moving right along, this is just the morphological analysis of PC3 cells. As we can see over there, for untreated cells, there's no visual, or we are unable to see that the cells were proliferate. We are able to see that there's no cell proliferation, and looking at the other two groups, we can see that the two drugs of interest were effective. And moving right along, this is just, these are the tumors after the xenograft model. As you can see that on cannabis, you can see the sizes of the tumors. We're able to compare that CBD and cannabis sativa, they do have an effect, but CBD is more effective than cannabis sativa. And then moving right along, over here we have the Western blot analysis. We're able to see that with treating the mice with cannabis sativa and CBD, as we can see along the lines there, the P53 is gradually highly expressed as you move along the different groups of interest. Now, in conclusion, the study was able to conclude that CBD serves an effective anti-tumor drug, and the treatment with CBD could be coupled with RBBP6 is more effective as compared to cannabis sativa extract, and the CBD were coupled with silenced RBP6 has the ability to upregulate P53 in PC3 cells, and the study further promotes the use of cannabis sativa CBD in cancer patients with an inactivated P53 gene. And lastly, the study recommends the use of CBD rather than cannabis sativa as an anti-tumor or anti-cancer therapeutic drug. And these are some of the references. Thank you.
Okay. She can forward the questions to Professor Mutadi because she has alluded to the fact that she won't be able to respond to the questions now. So this brings us to the end of this session for presentations, the oral presentations. The posters are still being evaluated, so I suggest the poster presenters make themselves available next to their posters. They are being evaluated, and the assessors will be going around to assess them. And again, I would like to apologize for the time. We had a bad domino effect. And I thank you all for your patience. You've been very patient and attentive. Thank you very much. Those who would like to join after the poster session, the ribbon cutting, and then after we will have a cocktail function in the same venue for the ribbon cutting. So everyone is welcome there. But the poster presenters, it's not over yet. We still have to go through the posters as well. Thank you very much for your patience and participation. Thank you.